Why we get started is to be mindful of time.
Sure.
And, I'm sure people will join as they go. So, first of all, good morning. I want to welcome everyone to our first virtual workshop for the Common Fund Data Ecosystem. My name is Noël Burtt. I'm representing the Knowledge Center. And I'm really happy you all joined us. I see some faces that I know, not faces, but names, whose faces I know, and some new folks. I wish we could all be in person, but we can do so much more virtually in terms of, spanning time zones and getting people who are in different geographic locations together. So today I'm going to kick us off before I introduce all of our speakers and kind of give us a little orientation to the Common Fund Data Ecosystem and start to talk about some of the objectives for today, because today's mission is really to share with you what the Common Fund Data Ecosystem is, to help you navigate its components, some of its data, its tools, its novel findings, which are the exciting part, and then really learn how you can use it in your own research. And so that's really the objectives today. It's a long session, but we'll have some breaks built in, so you can take a break. It is a webinar, but we encourage questions in the chat. We encourage you to raise your hand if something's really like burning. We're happy to do that. Mackenzie will be monitoring, and so will the panelists. We encourage you to look at this later if you want. We're going to record it. And we also encourage you to hopefully use the resources that we're going to share with you today. So I'm going to walk you through a little bit of all the speakers and what they're going to do, so you can have a sense of how it all sort of ties together, because really we wanted to think about this entire presentation as for someone who studies human disease. Really, that's the goal, and how the Common Fund Data Ecosystem can assist them or help them generate hypotheses or find data. And also, we really want to help enable discoveries with this resource. And what I love about the Common Fund Data Ecosystem is sort of like a beacon for the types of resources that, because there's so many resources in our field, the CFDE is really trying to bring many disparate resources together and make them more useful. So I want to talk about a little bit about that and give you a little orientation. So to orient you, to start you off to sort of the navigation of the ecosystem, I wanted to talk a little bit about the Common Fund. Normally, if I were in a room with you all, I'd ask if you ever heard about the Common Fund, but I'll make some assumptions here. I'll assume probably most of you have, right? And I don't know if anyone can name a Common Fund resource, but I bet you can as well. I bet you've used GTEx, for example, many of you, or you've heard of all of us that comes from the Common Fund. But maybe you don't know what the Common Fund data ecosystem is. And I think that's really our mission today, to describe all of those components to you and walk you through that. So from afar, what is the Common Fund? Just to give you a little orientation. And then Julie after me is going to talk a little more specifically. And I really wish our NIH colleagues could be here with us, because they've been such great partners in this effort with us. And we're really kind of channeling them today, because they really are champions for these funds and this Common Fund data ecosystem. But the NIH Common Fund spans NIH institutes, right? And it's really a bold scientific set of programs that are supposed to span biology, human health, and technology. And you can see them all listed here. There's a lot of them, right? And some of them are old, some of them are new. They tend to be 10-year efforts. But what we're really trying to talk about today is you may know some of them very well. Up close, you probably know GTEx quite well. If you study any genes, for example, you use GTEx to look at your favorite gene expression for your favorite gene and look at its expression patterns across an atlas of tissues, right? GTEx is one of the most used Common Fund data ecosystem resources out there, in my opinion. Another one, though, if you're an electrophysiologist, is SPARC. This stands for Stimulating Peripheral Activity to Relieve Conditions. And while they're trying to create networks of physiology responses across the body through electrical physiology responses, they also produce things like gene sets and gene expression data, which is far more useful to someone who's studying gene expression. And you may not be studying peripheral, signaling, but it could be relevant. So it's its own resource that's connected to the ecosystem. A third one is, if you're a biochemist, you might know this one. GlyGen is another example of a resource within the Common Fund data ecosystem. And this is for someone who maybe studied glycosylation. And this is a data aggregator that brings all the different data types across glycobiology together into one place so that you can analyze and use them. But what's great is they also produce things like gene sets linked to biological processes. So what I'm trying to get at is that while these resources are very niche in what they do, they also have broad application across sectors of biology. Another one you may be very familiar with is HubMap, for example. This is a resource to visualize the human cell atlas at a cellular level and allow you to access all the data therein. but they also have this huge consortium associated with them, producing results and making assumptions and observations from the data they're generating and making that publicly accessible. So it's not just a single data coordinating center that's producing a bunch of resources and data and making it accessible, but also resources all around them that are using that data. The ecosystem's goal is to enable some of that information to go to a broader community through coordinating things like common data models, common analysis tools, common visualizations, or creating novel findings from these disparate resources. So the goal is to create the ecosystem. About 6 years ago, they had a pilot. They brought all of these resources together. Some of them you know very well, some of them are new to you. They said, excuse me, sorry, that's a dog. And the mission was like to enable broader use of these resources to help you generate discovery, right? There are data sets that you may have never used before in resources you may never have known at, but there's data types within them in samples that may be useful, that could be useful in aggregate to put them all together. So as part of that, they created a set of centers, of which I'm a member of the Knowledge Center, to help bring pieces of this ecosystem to the broader research community. And we're going to talk through some of those as well today. But this is a complex environment of data, resource centers, and partnerships. So today's mission is to really walk you through all of that. But there are some themes and threads, right? So the themes and threads across these resources are, many of them are data aggregators. They bring data together from multiple resources or consortia that are generating this data into one place, harmonized and accessible, right? They also generate reference maps, right? Allowing you to learn a reference map for like links is an example of that, or GTEx, a reference map of atlas of the human gene expression, molecular signatures, observations from biological processes creating readouts, or gene sets, for example. So these themes and threads really we want to bring to you today because these are very relevant to you as you study human disease or biological processes. So our goal today, if we do well, is to help you navigate the system and maybe invite you and entice you to use some of its components, and also expose some of the really cool findings that have been done specifically by bringing data together across the Common Fund data ecosystems, data coordinating centers, and integrating other NIH resources. So you're going to hear about that today. So let me walk you through a little bit of that. So first, Julie Jurgens is going to take you through the knowledge, walk you through a little bit of the history, more than I did from the NIH perspective in terms of how the Common Fund data ecosystem was created, but then talk to you about the Knowledge Center, which is one of the centers to bring novel findings, to bring results from all of these resources together in one place as a place for you to sort of get a snapshot or a landscape of the types of scientific questions you can ask across the ecosystem. But what's great about that is it brings you to all of the other resources that generate the data or do the analysis that we actually put together in one place. So Julie's going to talk about that first. Then you're going to hear from one of data coordinating centers. While there's 18 of them, and this is the first of many workshops, we really want to focus on one specifically today because it was relevant to some of the story we want to tell. And David is going to talk from the coordinating center about MoTrPAC and its links to human traits. And what's great about this is it sort of gives you a sense of what a data coordinating center is because they have their own, resource and consortium generating results and research around the data they generate, but also how we as different centers are using results and knowledge from MoTrPAC to incorporate into other approaches and results that we generate as part of the Knowledge Center. So Jason's going to talk about that specifically. So one ample resource within the ecosystem are gene sets. They're produced as readouts for so many of these data coordinating centers, and they are readouts of biological processes or activities from these data coordinating centers. And what's great about that is if you put those all into one place, and you're going to hear about how we brought all those together shortly, you can actually link them to human phenotypes if you have another resource. And we are very lucky because we work with other NIH-funded resources that study human genetics of complex disease and even dipping into rare disease. So Jason's going to talk to you a little bit about how we did that. Then Oliver and Julie is going to come back and talk to you about how the ecosystem meets another environment. Terra. Many of you may be familiar with Terra if you worked with Anvil or Biodata Catalyst data or ClinVar, because many of the data sets in those resources are accessible through Terra. And really what we want to do with this is provide a new way for you if you are a Terra user or you are often accessing data through that mechanism. You might want to very conveniently access the results that we've got from the Knowledge Center. And this is just like an entree into bringing the Common Fund data ecosystem to other large-scale data aggregation resources and cloud environments to make it easier for somebody to navigate that space and make scientific questions and analyses. So Oliver's going to talk to you about that. Then we're going to have a short break in between. And then Avion from our Data Resource Center is going to talk about accessing all the data. So the Knowledge Center thinks about the knowledge and the results. The Data Resource Center thinks about the data and the metadata, because oftentimes you want to get at that data. So I'm just going to talk to you about the CFD workbench and some of the ways to access all of the data through the C2M2 model that the CFD has produced over the years, and metadata, and more importantly, some really excellent tools that allow you to access, analyze, and visualize some of the data within the ecosystem. As an example of that, and Giacomo is going to come on and talk about how we actually generate all of these gene sets. So many of these DCCs, I think 10 at least of them produce gene sets as outputs of all their results. Giacomo is going to talk about how they brought them all together and allow you as a user to find these, analyze them, and use them in your research. And that's sort of a piece of what the DRC does. And what's really cool is these gene sets we have used to link to human phenotype, which is what Jason was going to talk about. And finally, we're going to switch to other centers. Now, it's a complex ecosystem, right? There are 5 centers. There are 18 DCCs. You might be saying right now, even listening to me, gosh, how would I even look at this, right? But so Jennifer and Allissa are going to talk about how they've been funded very recently. They're just new to the team to actually train and educate you really well, better than we're going to do today at this workshop. This is more just an intro on how to use all of the data and tools within the resource and create really cool, mechanisms for education and training for the ecosystem. So you're going to hear about how they're going to do that. And then another center that just recently came online came from the Texas Advanced Computing Center. And this is our cloud infrastructure environment. So this is another way for CFD to also be accessible within another cloud environment. There are many cloud environments that data coordinating sensors access and use. For example, you have Cavatica within Kids First. You have Terra coming into the program. But the goal is to bring yet another cloud environment so that there is their interoperability and access across different environments through the open source for Galaxy. So what's great about that is you're going to hear about what John and his team are going to do to bring all of the Common Fund data ecosystem to another cloud infrastructure that's open access. And then finally, we're going to switch to a little bit more hands-on, get ourselves into the nuts and bolts of Terra. The reason we thought this would be valuable is because we're all NIH funded in many cases for these resources, and they are all over the place in many cases. And Terra is one really great place where there's access to many NIH-funded resources, whether it be the data, workflows, workspace. So Tiffany's going to talk about another ecosystem, which is Terra. And so what's great is that you'll hear from Oliver of how we've brought some of the CFD results to Terra through that workspace and what you can do in that. And then Tiffany's going to riff on that and bring that to, okay, well, if you want to work in Terra, here's other things you can do. More as sort of a tour of the different types of resources that Terra can offer if you are someone studying, disease biology. So if we're successful, we hopefully will provide you a framework for the goals of the Common Fund data ecosystem today. You'll have a basic sense of how to navigate the ecosystem, its tools, and its knowledge. And hopefully, most importantly, you'll be enticed to explore it yourself, because really this is about enabling discovery. And hopefully if we've done our job, we've brought the results and the data to you in a way that's easy for you to interpret and make sense of, and also ideally invite collaboration. And that's another thing to sort of think about. If there are things that you find exciting or interesting, particularly for us as a knowledge center, we're thinking about integration with external resources. What are the types of scientific questions we can ask with the Common Fund data ecosystem that we can use our incredible ecosystem of collaborators within the CFD to help us make accessible to you? So with that, I'm going to turn it over to our speakers. And I think I'm going to turn it over to Julie, who's going to be our first speaker today. And thank you. Julia, stop sharing. Are you good? Excellent. All right, I will go on mute.
Unmute myself. Okay. All right, so thank you, Noël, for that introduction. I'm going to jump right into it. But our goal today, as Noël mentioned, is to learn how you can obtain knowledge from the Common Fund data ecosystem. So just as a quick refresher, what is the Common Fund? As its name suggests, it is a funding mechanism, but it's really unique in that it actually supports multi-center, multi-institutional collaborative research. And in doing this, it catalyzes discovery and is able to address challenges that face not just one, but multiple research branches. So it can really transform research. As Noel mentioned, Common Fund support is designed to be time limited. So programs are typically funded by this mechanism and then obtain additional funding to keep delivering after that support has ended. But the Common Fund aims to accelerate emerging science, get rid of roadblocks that get in the way of research, build up the research workforce, and support science that is high risk and high reward. So again, to transform the research enterprise. There are three major types of Common Fund programs. The first of which perform sort of transformational science and discovery. So these programs identify new scientific discoveries, transform our knowledge, and identify new pathways to discovery. So they don't just do the discovery, but they pave the way for other programs to do something similar. Other CFDE programs provide catalytic data resources. So these programs help to manage and develop data from diverse sources for scientific discovery. And they do this not just by, say, storing the data, but by developing methods, standards, and commonalities across multiple programs that can be used in a reproducible way. Finally, there are programs that re-engineer the research enterprise. These change how we do research. They build up the workforce and they help to convert research from the basic research standpoint into translational and preventative measures. And finally, all of these types of programs are aimed to be compliant with FAIR principles and to promote open access sharing, reproducibility, and rigor within research. So that's the Common Fund and its programs, but what is the Common Fund data ecosystem? So the Common Fund Data Ecosystem, or CFDE, promotes accessibility, use and reuse, and integration of multi-program Common Fund programs. And it does this by building a cohesive ecosystem. It does this in a couple of different ways. So one of which is through formal partnerships. Examples of those partnerships are shown here. This was a recent partnership developed from individuals who lead the GTEx Consortium, studying transcriptomics, and xRNA, which looks at extracellular RNAs. And the goal of this partnership was to put on a workshop aimed at identifying novel biomarkers for extracellular RNAs. So this is a really nice example that's publicly available. It's recorded, so you can access it at this link here. And this shows an example of how two programs could partner together to develop a resource that wasn't available before. Similarly, the CFDE data distillery has come online to develop a knowledge graph. This knowledge graph includes data currently from 11 different CFDE programs and enables these really complex sort of queries across programs to look at relationships between gene expression, drugs, diseases, and other entities that are studied by each program. Deanne Taylor from the Kids First Initiative is heading up a sort of sub-aim within the data distillery, which looks at relationships between structural birth defects and pediatric cancers and their genetics and shared pathways. So this is a third partnership that has emerged more recently from CFDE. It just started in fall of 2024. But the goal of this initiative, this community data hub for integrated visualization, is to create a resource that lets you interact with, visualize, and explore predicted target genes of candidate cis-regulatory elements. So it does this by providing interactive sort of visualizations like you can see here, but importantly, it also harmonizes data, not just from Common Fund programs, multiple of those, but also from external programs like ENCODE. So this is an example of how these partnerships can extend beyond CFDE to really transform research. As you can imagine, everything that I've just described is very complex. So it requires a framework through which this ecosystem can stand and be supported. That's where these 5 centers come in. So the Data Resource Center enables users to search and use multiple data sets from different Common Fund programs. The Training Center provides training and outreach. We're going to hear from all of these programs today. The Cloud Workspace Implementation Center, or QWIC, provides cloud computing training and support. We at the Knowledge Center help you learn how to derive knowledge from CFDE resources. And then all of these efforts are coordinated internally by an Integration and Coordination Center. So we just talked about a lot of things. This slide is meant to just summarize the relationship between them. So the Common Fund funds these Common Fund programs, or DCCs, Data Coordinating Centers. A subset of these data coordinating centers are integrated into the Common Fund data ecosystem, both individually and through pairings that might have not existed otherwise. And these are supported by the efforts of 5 CFDE centers. We are one of these CFDE centers, the Knowledge Center. Our goal is to improve the understanding and visibility of CFDE resources, to generate knowledge by analyzing the CFDE data and providing it in a user-friendly and centralized way that people can access all in one go to explore other points in the ecosystem. Importantly, it's designed to be integrative and also focused on disease. Before we get started, I'm going to talk about how we define knowledge. As you can imagine, that's a broad term and it can encompass multiple things. And what we do at the Knowledge Center indeed encompasses all of these different types of knowledge. So it looks at foundational knowledge. So that's understanding the purpose of each CFDE program and its major deliverables, the primary knowledge or published findings from each program, secondary knowledge obtained from a single CFDE program through additional analyses, but maybe or maybe not published. And finally, tertiary knowledge. So these are assertions created by bringing in data from multiple CFDE programs. As you can imagine, the real power of CFDE comes through these secondary and tertiary knowledge branches. And so we're really going to hone in on unique offerings that we've developed through the Knowledge Center to create additional analysis and results findings. So there are 2 major things you can do through the Knowledge Center. The first is to learn about CFDE, and the second is to analyze CFDE data. I'm going to talk about each of these. To learn about CFDE, we put on regular research webinars. These are held on a monthly basis. This workshop is held in place of our research webinar this month. But if you say are interested in one and can't attend, or see that we have a previous webinar that you were interested in, we actually record all of these and have them available on our website, so you can check those out. You can also obtain CFDE program information. So through the CFDE website, CFDE Knowledge Center website, cfdeknowledge.org, you can do this in 2 major ways. The first is through this programs menu here on the top, and the second is through these individual icons in the schematic here. This is interactive, so you can actually click on a program, say MoTrPAC if you're interested in it, and it'll bring you to a summary page like this. So these summary pages have 2 different types of knowledge that we just talked about. Foundational knowledge, explaining the purpose, in this case of the MoTrPAC program, and major deliverables, and then primary knowledge. So that explains major findings that have come from the literature. And importantly, all of these have been curated in coordination with the experts from each of these centers to provide an up-to-date and comprehensive summary of what each of these programs does. So all of these programs are big, right? They do really important research and they do a lot of different things over the course of years. So I'm not going to have time today to talk about what every single one of these programs does. But this is just a high-level summary so you can get a sense of what the programs are and what some of their major offerings are. Glygen looks at glycosylation, or the addition of carbohydrates to proteins, and how that can affect various outcomes. xRNA looks at extracellular RNAs and their relationship to various phenotypes, as well as their use as potential biomarkers for disease. If you're interested in metabolomics, you should check out the metabolomics workbench. This is really a comprehensive one-stop shop for anything metabolomics related. For epigenomics and chromatin structure information, the 4D nucleome program is a great place to go. They have a lot of diverse data sets, as well as different methods and standards that they've developed. These two programs here, HubMap and Senate, have developed maps of the human body at the cellular level. And Senate specializes in a subset of those cells called senescent cells to explore their unique role in biology. Noël already talked about the GTEx program, but that looks at gene expression and regulation. And the LINKS program also looks at cellular and expression data, but identifies signatures from those. And it does this through various perturbation experiments. A2CPS is a new program that has come online recently that's looking at signatures in individuals who transition from having acute pain to chronic pain following surgery. So only some individuals develop chronic pain, and we don't really know why. So their goal is to identify both clinical, environmental, and also molecular signatures of that transition. Spark, Noël already talked about, but they specialize in the nervous system and in developing not just signatures, but also connectivity maps and an understanding of how neuromodulation can be used to treat various conditions of the nervous system. For drug development, Illuminating the Druggable Genome, or IDG, looks at certain classes of druggable proteins. So these are proteins that historically have performed very well and validated well in various drug development trials, but have certain understudied members. So those include GPCRs, ion channels, and kinases. And IgG helps to flesh out some of these understudied proteins to determine which of them could be drug targets. We're going to hear about MoTrPAC later today from David, one of our other panelists, but this looks at the molecular effects of exercise. Gabriella Miller Kids First looks at the genetics of rare pediatric disorders, and those include both structural birth defects and also pediatric cancers. And finally, this is another new program, Bridge to AI, but they're looking at integrating artificial intelligence into biomedicine. So that's not just through developing AI models, but also through developing rigorously assembled data sets on which to train the AI, building up the workforce, and developing standards for that. So they have a couple of sort of sub-branches. One of these develops clinical cohorts and integrates various electronic health records. A second looks at diabetes and how individuals have various signatures that change over time, and whether AI algorithms can be used to predict changes in those diabetes cohorts. Cell Maps for AI is another sub-initiative of Bridge to AI that looks at developing cellular networks that are AI generated from experimental data performed in cell lines. So these experiments target two sort of major classes of proteins with about 100 proteins in total. I'm sorry, 200 proteins in total. And they have various outputs in the form of multi-omics, imaging data, and more. And finally, there's a really unique and interesting subprogram within Bridge to AI that's actually looking at using voice as a biomarker for various diseases. So this is just a high-level overview of what these different programs are. And we can't talk about all of it today, but if you want more information, again, you should go to our website, cfdeknowledge.org, and sign up to attend our monthly webinars. So what resources have been generated by these CFDE programs, and how do you access them? You can do that on our homepage. So say you're interested in transcriptomics. You can hover over that word here, and it will show in this schematic which programs within CFDE have transcriptomic data or plan on having transcriptomic data based on their intended outcomes. So on our homepage, we've covered, sort of the major omics, but there are many additional types of data that are generated through these CFDE programs. So to learn about those, you can go to each individual program's website. For instance, here with Spark, you can see, yes, they have transcriptomics and spatial transcriptomics, but they also have developed all of these other types of data, phenotyping, biomarkers, microscopy, and maps. All right, so the second major thing that we do at the Knowledge Center is analyze CFDE data. So I'm going to talk about that now. The first type of analysis that we have available is by looking at genes and phenotypes and how they're represented through various CFDE programs. So this is an example of secondary knowledge that I talked about before, where we at the Knowledge Center take information that's been generated by these CFDE programs and synthesize it and analyze it in a novel way to bring unique insights. So this is the first type of secondary analysis that we have. You can search your favorite gene. This can be done through our homepage. So here you can see I'm typing in a gene name, and if you click on go to the KC Portal gene page, it'll take you to outputs that look like this. And these are all examples of different types of secondary knowledge that we've generated for that gene that was queried. I'm going to go through a couple of examples of those now, but you should really check this out because each has a unique offering and unique insights into biology. So this is a single cell browser that we've developed to look, in this case, at single cell maps of heart tissue generated by HubMap. So you can see interactive view maps. You can zoom in and out, toggle different cell labels on and off here, and look at gene expression, how it changes interactively within each of those cell types. This is another type of secondary knowledge that we've developed using Gabriella Miller Kids First data. So Kids First has performed DNA sequencing on various individuals, again, with rare pediatric disorders. And for understandable reasons, that information is protected within their framework. However, what we're able to do through the Knowledge Center is actually to represent that at an aggregate level. So here you can see for a particular variant, how commonly it occurs in various publicly available databases that we use for typical like population frequency controls like NOMAD, but also in other Kids First data. So if you click on this, sort of more information button over here, you can see within the Kids First data set, for each phenotype that was present, what the frequency of this particular variant was in the heterozygous and homozygous state. So you're actually able to do a form of burden testing for various genes and their potential and variants and their potential association with these rare phenotypes. So this is a unique form of aggregated knowledge that is not present on the Kids First website, but that we're able to extract through our analyses at the Knowledge Center. Similarly, we've produced a glycosylation tab. This is a form of secondary knowledge from Glygen that allows you to search by a gene and return a table of whether that gene is glycosylated, if so, what its glycosylation sites are, and whether it encodes a glycohydrolase or a glycosyl transferase. And again, this data is all available from Glygen, but not in this form. We've worked to synthesize it into a single searchable table that you can access in one go through our website. So we have similar sort of secondary analysis searches for phenotypes. Again, through our homepage, you can use our single search bar, start typing the name of a phenotype, say Alzheimer's, and it'll come up with a disease tab here where you can say view the KC Portal disease page. And that'll take you to a list of the secondary analyses that we've done for phenotypes. This is one example of a phenotype analysis result obtained from Illuminating the Druggable Genome, IDG. This shows potential drug targets for a phenotype with various levels of evidence that we've defined obtained from IDG itself. So you can see for Alzheimer's, there are these genes that have been predicted as potential targets. All right, so the other thing you can do in terms of analyzing CFDE data is really exciting. Now, these are all new sort of offerings that we have on the Knowledge Center, but we've developed these by analyzing data obtained from multiple CFDE programs. These are forms of tertiary knowledge, and I'm going to talk about each of them. So the first of these is a differential gene expression analysis. And what this analysis, you can access this analysis through two major places on our website. The first is through our menu bar up at the top here, and then the second is if you scroll down and click on the icon here. And that will take you to a page that allows you to identify putative biomarker genes whose expression differs in various tissues or phenotypes. So you can see the outputs for gene, tissue, or phenotype if you search any of those entities, and you can see what genes are up or down regulated in the different conditions. We currently have transcriptomic data that's bulk RNA sequencing from GTEx and Kids First, but have many additional CFDE centers that have produced transcriptomic data that we aim to integrate. This is an example of what the output would look like. You can see here a volcano plot showing differentially expressed genes and the same results in tabular form. Now, this is another tertiary knowledge example that we have. This is looking at sets of genes and their relationships to human phenotypes, pathways, and mechanisms. And this is a really unique novel analysis that Jason Flannick on our team, one of our panelists who's going to be talking soon, is going to go into in more detail. But it's a really unique approach because, as you can imagine, there are all of these knowledge graph assertions that CFDE and that others have been coming up with, and those offer a very specialized niche that has some pros and cons, and some of these are different from the pros and cons that are available for gene sets. So by integrating these knowledge graphs and these gene sets sort of separately, we're able to provide a really unique commodity within an ecosystem so that you can see evidence produced from both. All right, so gene set analyses enable you to perform cross-program analysis of these gene sets and how they relate to other genes, phenotypes, and mechanisms. I'm not going to go into detail because Jason's going to talk about that soon. But you can access this through our webpage, through this top menu bar, and again, by clicking on this icon here. Finally, we have some really exciting, hot off the press, newly available ways of engaging with CFDE knowledge. And that's through our CFDE Terra Workspace. You can actually interface with differential expression and gene set analysis results, and even bring your own results to this workspace. So Oliver is one of our panelists who's going to be talking about this in more depth soon, but stay tuned for that. And finally, I'm just going to talk about a couple of things that are coming soon. We're constantly developing new availabilities through the Knowledge Center, but these include transcriptomics for genes up and down regulated in response to exercise in rats developed by MoTrPAC, more transcriptomic data sets, more gene sets, and those are derived from both internal and external program data. All right, so finally, if you want to obtain CFDE funding opportunities, you can do that through this website here. This webinar is going to be recorded, so if you want the link, you can check it out there. This is one example of funding that's due at the end of February from the Human Virome Program, but these are constantly being updated, so you can check them out. So finally, we want to hear from you. CFD is designed to be a community and a community resource, and we want people to develop new ways of using all of the tools and methods and data that have been made available. So tell us what types of things you want, what types of trainings would help you, and what you use regularly so we can build these out more. So finally, I'd like to thank everybody at the Knowledge Center team for all of their hard work to develop these resources and more. And with that, I'm going to turn it over to Mackenzie, who will introduce our next speaker. Thank you.
Thank you, Julie. So with that, we are going to have David, who will be speaking on behalf of MoTrPAC, specifically elucidating the molecular effects of exercise. So,
Before we switch to David, actually, does anyone have any questions for Julie? You can raise your hand, and we'll pause for a moment, because I know we've thrown a lot at you in the short time. But again, feel free while David is setting up.
Feel free to use the chat as well if you think of anything later, and we can respond as we go through.
Beautiful.
and if you have a question and the speakers aren't available, we can bring that to them as well.
All right, beautiful. All right, David, let's hand it over to you. Welcome.
David, I think you're on mute.
Oh, yeah. I see. Still, I don't know how to use this, right?
Thank you.
All right. All right. So, thanks for the invitation. Hello, everyone. My name is David Jimenez-Morales. I'm bioinformatics lead as part of the MoTrPAC Bioinformatics Center, which is located at Stanford University. I'm thrilled to be here today to share groundbreaking insights into how exercise influences molecular systems across multiple tissues. The first thing that I want you to remember is that we are the consortium that studies exercise. Formally, MoTrPAC stands for Molecular Transducers of Physical Activity Consortium. And our aim is to study the molecular changes our goal is to study the molecular changes that occur during and after exercise. We are aiming to generate a molecular map of the effects of exercise as training. All right, why are we studying exercise, you wonder? Exercise is one of the most effective ways to prevent chronic diseases and promote overall health. Check out the extensive list of benefits. If you are not exercising, definitely you should. This is the best medicine that you can have. But very importantly, if you are a researcher studying any disease, consider this. Our data sets provide a reference, let's say, for the ultimate health state, right? So you can use to compare against, if you are studying any disease, to the findings in your own field, right? So we are committed to making this data available for the scientific community, for sure. And we are a multi-centered study, have many clinical sites. We have the animal sites. We have the chemical analysis sites, which are the labs that are all over the country generating the multiomics data. We have 10 labs, and then we have the consortium coordination. The Bioinformatics Center is part of them. So, I think that we are close to 30 different institutions as part of this project. And of course, and none of this would be possible without the incredible contribution of researchers, technicians, data scientists who make up the MoTrPAC consortium. And we are a multi-omics project. We are covering transcriptomics, epigenomics, proteomics, metabolomics, you name it, right, across multiple tissues. You will learn more about this as I go through the slide. Of course, we are a multi-species study. Our studies span both preclinical models, rats, and human subjects. In rats, we focus on acute exercise, which is a single bout of exercise that rats are around. We have endurance training. We do an 8 weeks training study. And we analyze 19 tissues, which is an unprecedented breadth of coverage. It has never been done before. We are conducting studies in both young adults rats, 6 months old, and also in middle-aged rats, which are 18 months old. And for the human studies, we are also currently studying acute exercise responses. We use, in this case, is cycling and resistant exercise. We are studying as well chronic training adaptation of cycling and resistant training. We have different cohorts, including pediatric, where we only do endurance exercise, and the sedentary adults, and highly active, but also highly active individuals. What are the tissue samples collected for humans? Is adipose, muscles, and blood and plasma. So MoTrPAC is aiming to address key gaps in the field. Here I'm representing a table with all the major studies, and as you can see, MoTrPAC is trying to increase everything. I'm trying to bring more knowledge if we can. So today I would mostly focus on one of the studies in rats. I think it's time to talk about the first study that we have been publicly released and published by the MoTrPAC consortium. That is about the chronic effects of endurance training in rats. If you got distracted, now is the moment to re-engage and pay attention to this part. I think it's key, because it will allow you to better understand everything else that I'm presenting after. So let me explain just the experimental design of this first study. I think it's just straightforward, but if you got it, I think everything else is going to make a lot of sense to you. All right, so for this particular study, we had, you see, these 60 rats, and 48 were randomized to the exercise group. And these rats every day are going to wake up and are going to do this bout of exercise. They are represented by each of this bar every day, and they are going to be training, and they are going to do this for during 8 weeks. We are going to be collecting samples, every 1 week, 2 weeks, 4 weeks, and 4 weeks. And then we have the sedentary controls that are not going to do anything, and that we collect the information after 8 weeks. Okay. So as you might notice, I'm not sure if you noticed, but the size of the bars here, the green bar, the sessions is increasing. It's getting larger, and this is because we are, interested in chronic progressive training, right? So we are increasing the inclination, duration, and speed of the sessions. But it's also very important that you pay attention that that we are interested in the adaptive training responses, right? I don't know what's happening there. We're interested in the adaptive training responses independently of the acute effects. So as you can see here, there is a little gap. What this means is that they are training during the week, then we gave some of the rats the weekend off, and then we sacrifice them 48 hours later. So because we don't want to account for any acute effect, we just want to see what are the adaptations in the system after being training for a week, 2 weeks, 4 weeks, etc. Right? All right. So let me give you a little bit more details so you visualize what we are collecting here. So again, as I told you, 48 rats are going to be running every day, and after 1 week, boom, we go and sacrifice the 12 first bout of rats, and we have a group of rats. We have 6 females, 6 males. And when we sacrifice these rats, we extract in the animal centers. They do this incredible job of extracting all these tissues. And for these tissues, we are going to do a number of omics assays. So we have for that we have like 10 different labs, as I told you. We have doing the transcriptomics, the epigenomics in the Stanford and Mount Sinai. We have the proteomics done at the PNL and the Broad Institute, and then we have metabolomics. We have targeted. And done and all these centers are listed on the screen. So here is more details. We are not doing all the assays for all the tissues, and this is because we didn't have a limited amount of funding. It would have been really cool, but this is how what are the assays, what are the omics that we are doing for each of the tissues. But it's in this table where probably we are summarizing everything. It's a better way to visualize what data we have, which are the number of training regulated features that we have. So it's a very unfair table. It's a very unfair table because it summarizes so many years of really hard work by many people. But as you can see, we are separating this by different domains. We have the epigenomics, so by the way, each column is a different tissue, and then we have the omics here. And we have the epigenomics, and we have the transcriptomics, proteomics, and the immunopanel and the metabolomics. So it's important to notice that in the background, the color that we use in the background is this gradient, which means that if it's dark, means that relative to the number of features that we detected, the number of differential features are relatively small. It's trying to give you that impression. just by looking at this table, you can see many patterns. For example, in the pigeon, you can see that we are detecting changes in the methylation and the chromatin accessibility, but it's relatively small relative to all the phenotypes that we are able to detect. But then, for example, in metabolomics and the immunopanels that we did, so we are detecting relatively a large number of feature type differential across all tissues. But then we have transcriptomics when we have a wide variety of responses depending on the tissue, which is really, really cool and very interesting. For example, the tissue that had the largest response was adrenal after the 8 weeks of training. You see there we have more than 4,000 genes are differentially expressed after 8 weeks of training. But then we have in the gastrointestinal tissues, especially in the colon, there's a lot of features that differentially express. And then it's followed by the white adipose tissues. Again, in the middle, we have all sorts of responses, and then we have the tissues that barely have any response, which are the brain tissues. We have the cortex, hippocampus, all those tissues that have a very low response. And so the same thing you see proteomics, we also have a wide variety of responses. And again, if it's white, it means that of course we didn't do the assay for that particular. Remember, for each of these boxes is focused on the 8 weeks, but of the entire training regulated features. But we have 4 time points for, let's say, each of the cells that are showing here. All right, so this is a massive research study, which was published in Nature in May last year. I encourage you to read it and find out all the details. It's an open source paper, open access paper. I mean, but I'm brave enough to give you the highlights for a very high altitude. Right, I'm gonna go to the International Space Station. I'm gonna try to give you the main highlights. So some of these highlights of the effects of endurance training are that there are immune system adaptation, less inflammation, let's say, or that's what we found. Optimization of the reparation and protection machineries, and of course, an enhanced energy production. And plus, I'm going to show you as well some interesting sex differences. All right, as you can see, let's go with the immune system adaptation. And as you can see in these figures, endurance exercise training kind of modulates immune pathways across multiple tissues. So it's represented, the tissues are each of the columns, with sex-specific and tissue-specific immune adaptation. One of the remarkable findings was the downregulation of pro-inflammatory cytokines in the small intestine, like, for example, CXCR3 and IL-1A, which were downregulated, suggesting like reduced immune cell recruitment and lower inflammation. And when it comes to reparation and protection, as you can see here, we were summarizing all the differential genes that are shared across the tissues that had a larger coverage of omics. And as you can see, there are some of the genes that are uniquely identified, differential expressed in these different tissues, but then there is a lot of overlap, different combinations overlap across the different tissues. And of course, something remarkable is this group here of 22 genes that are enriching all tissues and are related to, in general, to protection of the system, like chaperones. We have things involving protein folding in responses to heat stress. All that stuff. In fact, when we look at the protein abundance, we see how all these different proteins are heat shock proteins are increasing with as the training progresses.
David?
And then
we have a couple questions I thought was worth asking, just because it's nice to see the attendees starting to chime in. So one from the audience is, first, very impressive. Could you please explain what immunoassays and what is the aim to do the immunoassays? And in parentheses, they put for confirmation.
We have
question mark?
Mm-hmm. Yeah, we have an immune panel that we were where we were targeting a specific cytokines that we wanted to see the responses. We saw that this was very, we had these immune responses, and we wanted to specifically target some of these, some of the immune, some of the cytokines and some proteins related to the immune system that we wanted to specifically, kind of validate some of these findings. Yeah.
Very good, thank you. And then another one actually is from our panelists. Tiffany was asking, do the rats doing zone 2 training, are the rats doing zone 2 training, or are they working to max heart rate while running?
No, as 70% of VO2 max. Yeah, that's what we were aiming to. Yeah, thanks for the question. Yeah, I love it. All right, so the final highlight of this paper is of course, the enhanced energy productions, the mitochondria adaptations. Here, as you can see, we are showing a heat map of the differential abundance across the different tissues, the different the both sexes and the different time points. And of course, you can see that in skeletal muscle and heart, muscle and heart, you see how it's increasing the abundance of in mitochondria. The proteins are expressed in mitochondria. Then there are, with the exception of you see that pattern overall, but especially maybe it goes down in males in lung. And the same thing when we look at the protein acetylation, we had available for only these two tissues, heart and liver, but you also see that anything related to metabolism is also increasing in abundance. The extra aspect that I wanted to comment is the sex differences. As you can see here, we did the typical PCA, so when you get all the samples, in this case for transcriptomics, phosphoproteomics, and the protein abundance. And when we saw this originally, imagine this without the colors, it was like, oh my God, we have batch effects. What is this, right? So what we might have failed here somehow, right? But then when we start coloring this by the different groups, the different time points that we have, and by the sexes, it was clear that what we had is a strong sexual dimorphism in some of the tissues and for some of the assays, right? Here I'm showing liver, but I'm also showing a few, of these PCS for kidney, and the same thing for heart. After integrating all the data and doing this PCA, you see how it was important to we noticed that at least in rats, we could characterize these different responses depending on the sex. So after analyzing all the data, so many tissues showed these sex differences in their training responses, with around 58% of the 8-week training regulated features demonstrating some sort of sex differentiated response, which is really interesting. Again, this is on rats. We don't know. We are currently collecting and studying all the data from humans. Soon we will know if it's as strong this differential response in depending on the sex in humans, but in rats was really interesting. Just to let you know that we have, you can visit our portal, the MoTrPAC data portal, where we are making available all the data sets. You can access all the publications by the consortium. And of course, again, we are making available all what we are generating. As I told you at the beginning, we believe that we serve as a good reference state for any system. And that's why you can access the data through our data hub, but it's also available in public repositories. And of course, we are fully transparent, making all the source code available. And even more, if you are our user, we found that in our consortium, 95% of the people we're working on are, we generate the libraries where you just install the library, load the library, and you have all the normalized data, you have all the differential abundance results, and you can mine the data, do research on your own on this data, or most importantly, you probably can compare with whatever you have. So this is our goal. We're going to keep doing this for any project that we publish. Please come and check it out. So thank you very much. Of course, I want to acknowledge the people from the Bioinformatics Center, this magnificent team of colleagues that I'm so lucky to work with. If you have any questions, happy to answer any questions.
Thank you, David. Are there any questions from the attendees? There is one question. Is the human data still being analyzed?
Yeah, it's still being analyzed. The problem that we had is, I mean, this consortium started like 20, I don't know. So we started collecting data, I think it was 2019 or 2018. But the problem is that something happened in 2020 that made us stop the human studies. And unfortunately, so it took like 2 years to start all over again. But the data that we collected up to 2020, that data is currently being analyzed, is a cohort of 150 to 200 people that we are currently analyzing. That is still a lot of a decent sample size. Is we are gonna publish this very soon. We are currently working. We are writing the papers and working on making the data available. So in this year, for sure, that first set of for humans is gonna be released. And of course, we are working in acute exercise in rats in 6 months. Then it's coming the 18 months. So yeah, and then we will also analyze the data from the main study. We have 2,000 patients, 2,000 volunteers, data from all of them. The studies are bringing a lot of data as well that it should be very interesting and it should help us understand, how exercise impacts our molecular landscape, right?
Fantastic. Thank you.
Any other questions for David? All right, this is a good transition. How are we doing on time, Mackenzie? Are we good on time? Are we running ahead,
2
behind?
minutes behind, but we're okay.
All right, that's great. All right, so as we transition to Jason, I want to mention that, really the reason we had MoTrPAC talk is because a lot of what they do, they produce gene sets. And gene sets are a very quantifiable and measurable commodity that comes out of many of these data coordinating centers. And specifically, what you've heard about today is a lot of the data that is being generated is being made accessible, and that comes with metadata, and that comes with standards. And that's really an important commodity. But what we fail to sometimes appreciate is genes are very specific. And a lot of specifically MoTrPAC and other DCCs that Jason's going to talk about have generated a lot of gene sets. And we thought as a knowledge center, the story we want to tell today is how to take those gene sets, which are incredibly rich and actually voluminous quantities of data that the DRC, who you're going to hear from later, has helped aggregate. Jason's going to talk about how we can link those to human phenotypes. So with that, Jason, do you want to share your screen and we'll get you going?
Sure.
Beautiful. Thank you.
All right. Great. Yeah. So as Noël said, we're going to now be starting to take our dive into the data that's available within the CFDE with a particular focus on gene sets and their usefulness in understanding human disease. So that's kind of the focus here. And to kind of understand, I think everyone probably understands why the focus on human disease, but why gene sets? I think it's helpful to kind of think about this from kind of our perspective when we, as a knowledge center, we first started getting involved in CFDE. And the immediate challenge that you face when you're trying to integrate data and knowledge within CFDE is that the programs a part of CFDE were not started with like a coherent vision towards understanding disease. They all were started kind of in completely different areas of research. Many of them don't focus on human disease. So there's just this immense amount of heterogeneity. So I'm just showing here, like, the LINKS project, which is about linking perturbations to gene expression changes, the middle A2CPS, which is this study of a very specific syndrome. And then on the right, HubMap, which is like reference maps of single cell, gene expression. So these are all, kind of very different. And so linking them together, is it's not obvious how to do that initially and how you might kind of merge these data sets when they're not on the same people or samples or anything like that. So when you start to, I think, as human geneticists, that's our background. We typically think of human disease and, not environmental or infectious diseases, but, genetically influenced diseases as this chain of events from a DNA mutation up to alterations of gene regulation in certain cell types, and then ultimately higher level cellular processes and biological processes that ultimately underlie disease. And kind of on this framework, you can start to think about what do these common fund programs tell us about this kind of pathway to disease, and kind of just a back of an envelope, like, survey of what type of data these projects have. A lot of it really focuses downstream on genes and their functions and human and human phenotypes. Okay, so really that's the area that we're going to try to be using CFDE data to inform upon in the context of disease. Not really as much like specific molecular changes and regulatory changes, but genes and processes and functions. Okay. And so, I think the MoTrPAC was like a great example of this, right? That's a study of a very specific, I don't know if you call it phenotype, but a human activity and its influence on a variety of phenotypes, and ultimately trying to identify underlying molecular changes and linking those to processes. And so that obviously could be of high interest to many people studying traits that are influenced by exercise. And you could start by looking at the publications that, David did a great job of just going through the highlights of that. But of course, that's only a fraction of the information. And our goal is to really unlock everything that's in the data for people to make use of. So one approach I just want to mention that a lot of people are taking now, and it's gotten a lot of attention within many fields, particularly NIH, has been to start to try to express the information in these data sets within knowledge graphs. It's kind of this very general, flexible way to encode various different aspects of data and knowledge within these assertions and entities. And it's this very abstract and general way to model this. And that is something that actually is available within the CFDE, and Jonathan Silverstein and Deanne Taylor are really leading that. And if you are interested in learning more about this, we will be happy to talk with you about it, but it's not really the focus of what we'll be talking about here today. Instead, we're going to talk about kind of a complementary, or I mean, not really complementary, but kind of orthogonal, different way to kind of join across these various different programs. And that's, as Noël mentioned, this concept of gene sets. So really a gene set is just any collection of genes that can be generated from pretty much anything. And because they're so general, you can generate them for almost any CFD project. And I'm showing here an example of what this would look like for an example MoTrPAC. This is from the DRC. These are genes that had expression upregulated after 8 weeks of exercise in the blood of, I think it's a specific rat, although maybe David can correct me if I'm wrong. And you get, I think, 689 genes that were up. And obviously you could go into more detail about, the degree to which they're up or down regulated, but for now, we're just going to consider this to be, this is the collection of genes. And you can imagine doing this really for any type of process or any type of data. There's many things that can be done to generate these gene sets. And really, Avi Mayon's group has been doing this, who's one of the PIs of the DRC for many years. And so this is not a new concept. And because it's not a new concept, that's why we think it might be a really valuable way to kind of link together these different programs in CFDE. And in fact, there is a resource of about 37,000 of these that you can access through the DRC. There's also tools, I think Giacomo is going to describe some of them, that help you manipulate and union and join and intersect these and augment them with your own gene sets. And so there's a bunch of infrastructure that's available for generating these within the CFD. And we're going to be continuing to do this activity and augment it further in the coming months. I think you'll hear more about that later on here today. So the other end of the sword, though, is that because you can generate so many of these, it's actually not trivial to kind of make sense of the gene set libraries that you have, right? So just because I can generate a gene set doesn't mean I can understand what information it's capturing, right? The information that you can think of a gene set as almost like this noisy readout of some biological process, right? That depends on the data and also your method for constructing it. So everything's implicit. Some of them are junk, right? They're just not actually tagging anything. And also like, just because it captures some process doesn't mean it's relevant to a human phenotype or a disease of interest. So really this task of sorting through this huge library of gene sets and figuring out which ones are interesting and which ones we want to focus on, the canonical approach that people do are through gene set enrichments, which basically means you take any input gene list or gene set and you cross-reference it across a bunch of gene sets with known biological functions. And when you find significant overlaps, that gives you insight into what your gene set is tagging or what it's doing. And that's really the goal of this workflow that we'll be talking about, and and Oliver will be going through some demonstrations, which is to try to use this concept of enrichments to link these CFD gene sets into processes tagged by human phenotypes, okay? And really producing these relationships amongst genes, phenotypes, and then these pathways, which we're considering gene sets to be a proxy to. And so the method that I'll very briefly, I'm not going to go through the details of this, but we do have a method that's currently, it has not yet been published, but there are a couple webinars available that you can watch on our website to see in more detail how this method's applied. Here, I'm just going to kind of today go through how to interpret its results and kind of motivate a little bit why it's slightly different than the traditional enrichment approaches you might be familiar with, just because that kind of basic background's necessary for understanding the output files and the workflow that Oliver is going to go through. Okay, so let me see, is there anything in chat? No, okay. So let's begin with like kind of the basic classical enrichment test that many of you are probably familiar with. So the typical way that you would do a gene set enrichment is you have like some, and I stole this from some website, so it's not necessarily clear in this figure, but you have like a set of genes of interest that's, you know, some subset of the universe of genes. You have a known gene set, or, you know, which has a subset of genes in the universe in there, and you do this basic like hypergeometric or Fisher test to try to see whether the overlap is significantly greater than expected by chance. And that's kind of the normal way gene set enrichments are done. When you think about now doing enrichment between a CFDE gene set and a human phenotype, right? The human phenotype, it's not, you have to first generate the gene set for that human trait, right? And one way you can do it is you can kind of like go and take the genes that have been associated with that trait from like the GWAS catalog or like the human phenotype ontology, but you really throw away a ton of the data, right? In reality, what we have from human genetics for a trait is like variant level association data, which needs to be mapped to genes and used to create like some substrate with which you can do that enrichment. And for those who've, you know, familiar with human genetics, like a very, you know, the way that this is typically done, and MAGMA is the most popular method for this, is that you take the full list of associations across every SNP in the genome, and you get these quantitative score for every gene in the genome. And then you kind of like regress that score on your gene set, and that gives you these p-values. And these are very, very, that you can then use to, you know, evaluate which gene sets are enriched for that trait. I'm kind of showing here what that output typically looks like for, here's like an example trait against some not CFDE gene sets, but the MSigDB database. You can see you get these p-values. And this is like very, very widely used, and it's commonly, you know, applied to many genetics association studies. But it has some limitations, which is especially for our goal of kind of sifting through all these gene sets that we're generating for CFDE. It's kind of very hard to interpret what a p-value means. And there's also tons and tons of associations, in particular amongst gene sets that are like highly correlated, right? So like abnormal waist to hip ratio, the genes in that gene set are probably very, very similar to the genes in insulin resistance, but both of those are very significant. So you end up with tons and tons of significant associations. So the main use case of this method Pigeon, which again, I'm going to go over very briefly, is you can think of really two main advances over MAGMA. One is that rather than generating probabilities, it has much more interpretable coefficients that can be related to probabilities, which allows us to kind of just more easily use the results in downstream analyses. And then also very important, it's a very, very sparse model. So the vast majority of gene sets linked to a trait get completely ignored, and only a small number that remain have more interpretable coefficients. And we'll see examples of that in a minute. What exactly I mean by that. So first, let me just again, this is mainly for people who are just interested in the statistical details. You don't need to follow this, but just to give you a high level, the basic idea behind Pigeon is that it's an explicit model as to how gene sets affect the probability that genes are involved in traits. And then it also has a model within there of how genes involved in traits affect genetic associations observed. So if you're familiar with graphical models, this is what it looks like, and there's all these equations. But basically, the high level is that you pose some model, and this is the sparsity here, that most gene sets have absolutely no effect on the gene's involvement in a trait. And then those that do kind of add to affect the probability that a gene is involved in a trait. And these nodes up here, you can think of, there's like 20,000 of these, one per gene. There's one here for all like 150,000 CFD gene sets. And then you impose some model about how this is the SNP level data up here, how that's affected by the involvement of genes in disease. And that whole model is this kind of Bayesian model that allows us to do inference from the genetic associations backwards to estimate these quantities. So intuitively, this is the intuitive interpretation. You have these like initial noisy guesses as to which genes are involved in the trait you're looking at. You group those that are in gene sets, and those are supposed to be the blue circles. And then based on your initial guesses of what genes are involved in the trait, you kind of identify the gene sets that are enriched for those, and you kind of then re-estimate. Now the genes in those gene sets get higher probabilities. That changes the gene sets. You kind of go back and forth until you get the model converges and you get these quantities of gene probabilities and gene set effects. Okay, and basically these are the point where these are the key quantities that we're going to be kind of using throughout here. So you get gene level values. Okay, I remember I had that triangle of gene set, and trait and phenotype, right? So all this is anchored for a trait. For every gene, you get a probability it's involved in the disease. And that actually is decomposed into the probability coming from the genetic data observed for it. So that has nothing to do with CFDE. That is just based on like the observed genetic associations for it. And then you also get how much that probability is influenced by all the annotations it has, right? So that to some extent is relevant to CFDE because the genes that have the highest indirect support here are those that are going to be in CFDE gene sets that are enriched for the trait. And the CFDE gene sets enriched for the trait have these values that tell you how much they affect involvement of a gene in the trait. And those you can think of as the analog of like the strength of enrichment. And what's nice here is that you get not only the marginal value, and for those who don't know that terminology, that's if like you run a model with one gene set for enriched for a trait. That's typically what like all the other enrichment methods do, right? That's why you get very low p-values for two highly correlated gene sets. But in this model, we also have joint effects. So we can determine the effect of the gene set conditioned on all of the other gene sets in the model as well. So you can kind of have this nice breakdown of, okay, this gene set is enriched for the trait, but it's not like independently enriched for the trait after correcting for everything else. I think it's easier to understand kind of a concrete what this looks like. So this is not using CFDE gene sets. This is like a bunch of like known gene sets from the mouse informatics database and text mining. You don't really need to know what these mean that much, but you can probably guess. And here are these and this is for type 2 diabetes. Here are these gene probabilities. So as I mentioned, you get the direct and indirect. So these are, insulin, if you're familiar with diabetes, right? That's got very strong support from the direct genetic evidence. It's also in a lot of gene sets that are enriched for diabetes. And so overall, it's almost certain to be involved in diabetes. And then over here, again, you have your joint and your marginal effects, right? So that's what I mentioned. These are the effect sizes of the gene sets in isolation. This is the joint model. And you can see there are examples, in most cases where a gene, let's say like exenatide, has a decently high marginal effect size, but a much smaller joint effect size. And as we're going through, just some ballparks to keep in mind, these numbers may look small, but they're in units of log odds increase in probability. And so anything over 0.01 is actually significant. And anything over 0.1 is quite significant. Anything over 1 is like extremely significant. So even though this like 1.2 looks very small, that's actually very, very highly significant. And then over here, these are units of probabilities, right? So we set a background probability of 5%. So obviously something that's at 67% is quite high. And also on the portal, what you'll see and on the data, a lot of the probabilities are in units of log odds as opposed to probability, but it's kind of all part of the same thing. So don't need to worry too much about the units at the moment, just to give you a sense if you're wondering like why these might look quite small. And then just kind of illustrating, this is the total number of coefficients in this model that are, and you can see the distribution. There's a very small number, and they're all positive. And I'm comparing this to, if you are familiar with GWAS and this approach of using gene sets to prioritize genes, you might be familiar with this method called POPs. It's very widely used. And you can see here, by contrast, its model has a lot of non-zero coefficients that are all positive and negative and much harder to interpret. You can kind of see that here. I'm re-showing like these gene set values. They're all positive. There's only 100 that are non-zero. Whereas when you're trying to look at running POPs on the same model, you can see you get just much harder to interpret, right? You have marginal effects that are negative, meaning that they're actually depleted for genetic associations in isolation, but in the joint model, they become positive. And so that's just one of the byproducts of using models with many, many gene sets as predictors. And that's one of the main reasons we're using this tool, Pigeon, here rather than something like POPs, because our goal is to really try to identify the very small number of gene sets that are enriched for a trait in this CFDE kind of workflow. Okay, so what the resource we have is basically applying all of these CFD gene sets, and there's about 140,000 of them, which include all the ones I mentioned earlier that are in the DRC, significant pairs of gene sets that are overlapping, as well as some of the differentially expression gene sets Julie mentioned. And we've cross-referenced those with 1,200 traits across our knowledge portal that we maintain for NHGRI called the Association of Function Knowledge Portal. And again, like, you can think of these as these triples where each result is gene phenotype pathway / gene set. So I'm going to go through just kind of in detail, like one example, which I hope will kind of crystallize like how to interpret these and what to use. And then you can browse all of these at this URL if you're interested. I'm not going to go in too much detail about how this works, but you come here. And there's basically just this giant table of like every triple, right? So the way to read this is like, here's your phenotype, right? Here's the gene set, and here's the genes in the gene set. There's 19 of them, and this will kind of pop up those that are involved. I'm going to go through in detail how to interpret one of these examples. So in a minute, but you can, through this website, you can click on genes to get more information. I guess I'll do an example of that. Now you're kind of anchoring these results at the gene level, so you can see all of the phenotypes that gene's associated with. And then by contrast here now, all the gene sets that it's associated with, right? So on the previous, so you can, again, everything is about these like triples, but you can kind of anchor to a gene set, you can anchor to a gene. If you want, you can anchor to a phenotype, and you can see like all of the top gene sets associated with the genes, with the phenotype or the genes. And so that's kind of the easiest way to browse and explore this resource, right? But today we're trying to talk more about how you can like kind of interact with it in like a workspace and download the data and really do more informatics stuff beyond just the portal. But if just, for getting familiar with it, this is probably the best way to do that. Okay, so let me go through like one specific example. All right, so this is from MoTrPAC. I think it probably makes sense to stick with that since we just had a presentation on that. I can't remember how I got to this. I think it might just be the top MoTrPAC gene sets on that page. But so here is a relationship between reticulocyte count, the gene set T30 blood RNA male 2 weeks up, which, so one thing we're working on is documenting these gene sets better. But this is genes upregulated after 2 weeks of exercise in the blood. And then there's 184 genes in there. And if you expand, these are the top genes. All right, so what does that mean? So as I mentioned, that's the gene set, okay? What we are showing is that the phenotype is reticulocyte count, which if you're not familiar with that, that is a count of immature red blood cells. The marginal effect here is 2.06. As I mentioned, that's very high because 1 is extremely high. That's the increase in log odds of a gene trait relevance for genes in the set. And then the joint effect is 1.38. So this actually not only strongly associated with reticulocyte count, but even after controlling for all the other gene sets, it's quite high. There's 184 genes in that set. And one thing that's really critical here is not all of these 184 are relevant to the trait, right? There's only a subset. All this tells you is that it's over-enriched, this gene set, for genes associated with this trait. So if you want to know the specific driver, quote unquote, genes, you expand this table, and that's where these genetic support that I mentioned, direct and indirect, can kind of tell you which are the genes that have the strongest associations, right? And so you can see, I'm just kind of highlighting here, by default, it's sorted. But if you went to the very end, you'd start to see things that are in the gene set, but have very low, like actual associations with reticulocyte count. So inference possible here. So first off, we filter down from like 137,000 gene sets to 1 that is enriched for this trait, right? So this trait is of interest, right? Almost by definition, that it is associated with some human trait, and it tells you which human trait that is. It then gives you the specific mediators within the gene set that you should focus on, right? Again, like you can think of a gene set as a tag of some noisy process, so it's going to include some things that are not relevant. And then also critical, if you're not familiar with genetics, this is actually a causal link between these genes and the phenotype, right? This is not like these genes are upregulated as passengers to exercise in response to reticulocyte count, right? A lot of the genes in the gene set might actually just be passengers that like are upregulated in response to exercise. But we know that these genes actually, if you alter them with genetic variants, they causally affect reticulocyte count. So these are almost like driving that phenotype that you're observing. And so that's kind of the total of the inference here.
Jason,
yeah.
might we interrupt you for a second? This is a nice question in the chat. The attendee apologizes that they missed this, but is Pigeon using a similar assumption to Magma of combining SNP level statistics into gene level scores based on physical proximity of the SNP to gene bodies?
Yeah, very good question. So that's all encoded in this map between gene to SNP. And right now, yes, it is basically a byproduct of, it uses a kind of a decaying window, similar to how Magma does, but with slightly more like fine mapping of genes, of variants in there. We have run models where it can use like SNP to gene links as part of that and not just a fixed window. But all the results we'll show here today, you can think of, it's fine to think of as for each gene, estimate its probability based on SNPs nearby, and then you refine that using the gene sets. And like I said, I'm happy to follow up more about the details of that. There's substantially more in there, but that's a perfectly good assumption for today. Okay, so like that inference that I mentioned here, you probably were not surprised by this because red blood cell production is obviously very well known to be involved in exercise training. I'm not an expert in this. Maybe David can comment, although his group did suggest this example, so I suspect it is consistent. One thing that, again, I don't know if this is of interest, but at least to my knowledge, usually it's erythropoiesis, which is the main mechanism through which these red blood cell count is supposed to be affected. But in this gene set, you actually see some other, and you see that here for some of these genes. But there are other genes that have other roles in red blood cell, like transport and integrity and stability, that I have no idea whether these are known mechanisms. But hopefully this gives you a sense as to how much information you can get from just this one resource, right? And there are many others. I'm not going to go through these because I want to keep the time, but maybe I'll just, here's an example of a gene set from Lynx, right? So this is a gene set that are genes that are upregulated after treatment with this compound. Exact same inference here, right? 239 genes. Here's the genes. Marginal effect here is a little bit lower. You can see here, this is one that the joint effect is quite low, but it's still the genes in the process are associated with the trait. And this is actually an example where if you look up this compound, it's not under development for inflammatory bowel disease, which is the phenotype I showed here. But it does have, various properties that are actually consistent with what we know about IBD. So not necessarily something that you just would immediately go to investigate this as a treatment, but certainly kind of hopefully giving a sense that the biology makes sense. And there's other examples here I'm going to skip because I want to get to one other, so kind of a use case that Oliver go to in a minute. The first one, or maybe be the second one he'll talk about, is simply taking that gigantic table of all gene set, gene phenotype associations and kind of cross-referencing that with genes that you may have as a user to kind of like look across these pre-computed tuples. And he'll go into much more detail about what that is. That's probably pretty obvious about what you could do. One thing that's less obvious is that GWAS associations are not actually required to run this approach, right? So we just, as I just mentioned, currently uses this mapping from SNPs to genes to get their probabilities, kind of like Magma. But if you know gene probabilities just directly, you can just run the model based on that. So this is an example of like a rare disease where you kind of just know the genes involved in it. I'm showing this for a rare form of diabetes. So let's say there's 10 genes that you just know have very high probability of being involved in MODY. All the math and everything else remains the same. You're just not using that SNP data. You just directly know the probabilities. And you get the same thing back, right? You have the genes up here that you can see I've encoded these as having a 95% probability. Those are all the known MODY genes. But then you kind of fill in additional genes that are involved in the pathways enriched for MODY, right? And you get these combined scores, and then you learn the actual, all of the pathways that are involved, that are enriched for your gene set, right? So you can think of this as like almost a very stringent, interpretable way of doing a gene set enrichment of your input gene set across a huge library. Again, so no new idea, but a new method, right?
Jason, I have one more question from the
Yeah,
chat. Is it
yeah.
possible to detect co-expression between two specific genes in a particular disease like leukemia, for instance?
Is it possible to detect that? That would probably be upstream, I would think of this, which is you would analyze co-expression data to create gene sets that would then go into the model. The output of this method would not be able to tell you anything about co-expression, but we do have in the CFDE a lot of gene sets that have been created. So Avi's group has this resource called Arches, I think, that basically systematically combines a ton of gene expression data to identify kind of networks of genes co-expressed. And there are a bunch of those. I think I even had an example earlier where you can now cross-reference those with traits, and that helps you zero down on co-expression relationships that might be relevant to disease. But it wouldn't like produce novel co-expression relationships. That's kind of upstream. But maybe well, I'm not sure if we'll talk about later today, but it's definitely something that we are going to be generating using CFDE data. And so just to conclude here for this second use case, this is where, I think Oliver will show in Terra, you'll be able to input your gene list. You can then run this method using a WDL that will essentially run the full model to produce all of the outputs that we just went through, but now tailored to your list as quote unquote the trait, right? So rather than seeing creatinine kinase associated with this CFDE gene set, you will see all the CFDE gene sets that tag your trait. So it's quite your gene set. So it can be quite useful for trying to get insights into the processes that are tagged by CFDE that might give some information as to what your gene list is capturing, and also predict some additional genes that might be candidates to augment your current gene list. There's other ways that people do that with like literature links and co-expression, but here you now have a rigorous probabilistic model that will both predict the probability that genes in your gene set are relevant to the underlying biology it's capturing, but also find additional ones. And I think Oliver is I'm hoping I'm not selling something Oliver won't do, but I think he's going to go through both of these two use cases as to how to make use of these data. And so, as I mentioned, we have about like 1,400 of these traits. I guess it's gone, maybe it's only 1,100. I can't remember the total number. Coming next week is going to be adding many, many more traits that will have not just from the NHGRI report I mentioned, but pretty much everything in the GWAS catalog, even things that do not have full summary statistics available, and a bunch of rare diseases. So we have not looked at that data yet, but it has been run, and we will put that up soon too, so which will give you many, many more human phenotypes you can kind of work out with. And then I think Oliver was going to go through these files, but there's basically 3 types of files that have the gene level statistics, the gene set statistics that I mentioned, and then finally like kind of which for each gene, which gene sets it's in. So that kind of like completing that triangle, and we'll have that for every trait. Okay, so that's it for me. I'm not sure if there are any other questions, but if not, I think, are we taking a break or is Oliver up next?
Oliver's speaking next. Yeah, go ahead, Mackenzie.
Great.
I was just going to say that.
But Julie's, I think, going to frame it for us.
Okay.
But I'm curious, Jason, and I think maybe the DRC is going to talk about this later, but because these gene sets are like living things, do you have any thoughts about how we might, as a community, not just CFDE, create a mechanism? I know Oliver's going to speak to this a little bit, but to make it easier for people to curate and submit gene sets more broadly and almost create a paradigm for gene set collection across different NIH funded resources?
Yeah, so that's an interesting question. So the first thing I should caveat is that I am not a gene set analysis expert, right? So I'm a human geneticist. And so I've kind of come into this gene set field like the original goal of this pigeon method was to prioritize genes for GWAS. But we've, I think, realized over time how fundamental gene sets are for understanding biology. And I'm only mentioning this because Avi, in particular, and his group have been doing this for many, many years and have already done an incredible job of curating from public sources tons and tons of gene sets and might be better qualified to talk about the challenges and the process and what's already been done. Having said that, I do think that particularly in the context of NIH-funded projects that we're involved with, I do think this is significantly exploited. And Avi, I know, agrees with me on that too. And I kind of frame this intentionally that I think there's been a lot of attention for knowledge graphs within these resources, which are very important, but require a lot of manual curation. And gene sets, especially I'm hoping with the method that I talked about today, you can kind of be more casual of just gathering everything you can and then rely upon the model and its like severe sparsity constraints to kind of filter them out, right? That's like a very common technique in machine learning. When you have tons and tons of predictors, you can kind of let the data find the ones that are important. So building up to your final question, I do hope that we can demonstrate with by maybe doing this with CFDE, extending more broadly, that maybe some community efforts to systematically submit and gather gene sets. And I mean, there's already, like, Avi maintains a tool Enricher that people submit those to. I think by demonstrating the value of maybe you get that, you can cross-reference it to human traits, you can cross-reference to these libraries, it's really useful. We can start to maybe do a more systematic collection. But again, that's something that Avi, I'm sure, has thought about and might also be able to give more details on the challenges there and what's already been done in that space.
I love it. And also, I think our job, as human geneticists, resource builders, is just continue to curate the human phenotypes. And I think, as you alluded to, right, bringing in the rare disease component into the results that we have from the human genetic side and pairing that with a more concerted effort in gene sets will really make a resource that will be valuable to, a broader community of researchers. And with that, I hope that Avi and Giacomo are listening right now, because I think we really want to hear a little bit from you as we go to the DRC later in the session about how you are thinking about the extension of some of the work you're doing with the gene sets Enricher. So with that, is it Julie now up?
Yes.
All right, fabulous. Any more questions for Jason, though, before we turn it over to Julie? All right, lovely.
All right, thank you, everyone. So you've just heard about two really exciting forms of tertiary knowledge that we've generated through the Knowledge Center. So these are the gene set analyses that Jason just mentioned through the Pigeon method, and the differential expression analyses looking at transcriptomic data and what genes are differentially expressed in different phenotypes in CFDE. So you can access these obviously through the mechanisms we've just talked about, so through our website, cfdeknowledge.org. But what if you want to do more? So what if you want to further analyze these data? What if you want to connect them to other resources or analyze other resources in the same way? What if you want to bring your own data and run one of these analyses on them? Oliver is going to tell you about a Terra workspace that he's developed, which enables all of this. So this workspace is available through a public URL. I'm going to share it in the chat momentarily, so you can access it. But in the meantime, I wanted to emphasize that this is really a community effort. So as Jason said, this method is brand new, and he is actively developing ways of integrating new gene sets and new ways of making this like a community resource. So if you have ideas, if you're interested in contributing gene sets or in validating this method, please contact us at cfdeknowledge.org. All right, with that, I'm going to turn it over to Oliver, who's going to talk more about what this Terra workspace is, how it works, and where you can access the data. Oliver, take it away.
Hi, Heading. Okay, you guys can hear me? Okay, good. Let me share my screen, especially, yes, the workflow. Okay, now you should all be able to see the workflow. Like, sorry, not the workspace, yes, on Terra. Thanks for Julie, also for beautifully setting up the description of the workspace, which you can see here with graphics and everything. And what's a workspace? It's basically a space to share data and workflow. So we have data, various data. We have some gene expression data, which I'm not too familiar with, but just point out there's some from GTech. And there's also some from Kids First. And what I'm more familiar with is, of course, the pigeon output. So as Jason described, you get relationship between genes and gene sets. And we have pre-calculated results from like 1,300 different traits. So there is, for every trait, we have gene statistics, which you can look at here. This is how it looks like. So there's gene and different data about that gene. And then we have gene set statistics, which look like this, and then the gene set and a few other data. And then there is, of course, the connections between the genes and the gene sets, which are here in gene set stats. Yes, and this is how that looks like. So, and then we also we have a table of all the data. So here we have a table phenotypes. So we have all the phenotypes, identifier, a description, what they are, and for each we have a link to each of these three files: gene stats, gene set stats, and gene and gene to gene set stats. So, and yeah, if there are any questions, feel free to interrupt. And then we have workflows. So one workflow is to actually run Pigeon yourself, and the other is to filter these by genes. So let me first so for either, we start with a file that is just a list of genes. Let me here. So this is a file that you would upload to the Workspace, and that has a list of genes. I created a list just randomly with those 5 genes in it to be used. And then you can use that to filter one of the files that has a gene in it, which is basically either the gene stats or the gene set stats. So for example, so this, I call it grab genes. This one you can use to filter. So basically, you want to have a link to the data file that you're using. So you can browse the Workspace or point to any other file that you have access to. And for example, you can pick here from gene set stats. You can pick any file that you like. For example, I just randomly pick one. Let's say this one. And then you here is a link to the list of genes that I just showed you. And then just a name for use as a prefix for the output files that you will be getting. And yes, and then you say save, and then you can run the analysis. Yes, that's one way to do that, where I basically point manually make sure that the right file paths are included. There's also another way of running workflows, which is by using data tables. For example, let me go back to the workflow. I say here, run workflows with inputs defined by data table. So I select my phenotype table, and then I can select rows from that table. For example, any rows I want. So for example, that I could pick those 3. And then, okay, that's my selection. And then I can do the following. I can, where it says data, I can say this, and then point to fields in the data table. For example, here, this gene stats, I choose this one, and then it will pick the files from the data table. So I can save this and then run it, and it will run this for each of the 3 rows and use the linked file. Yeah, so this is the workflow to filter the data by your gene list. And the other workflow we have is to run Pigeon yourself, where you also use a list of genes. Basically, I use here a link to the same list of genes that I use, and then give it some name as a prefix for the output files. And yeah, and that's it basically. And for example, if I change this, I need to save again. And then I run the analysis, and that will just run Pigeon on your list of genes, and then eventually get the output files. Now, ideally, the output file should appear here under certain conditions that I'm not sure. But you can also, if I look at for example, if I go to the job history, I look at previous runs that I did. Here, for example, where I filtered some genes, you can go to the execution directory and you can go to the task that filtered, and then here I have the file with the selected genes. And I can download it, for example. And same thing for, if I go to the job history, same thing for running Pigeon itself. I can go to the execution directory. And yes, and the files are here. There's gene set and gene set starts, and gene stats. And I can download any of those. Yes. Okay, yeah, that's mostly what I want to show. Yes, it takes a bit for the workflows to actually run, because it has to commission the compute node and upload everything to compute node and then run it. And then download the files back to cloud storage. Yes, are there any questions?
Thanks, Oliver. That was great. Just to kind of summarize for everybody, do you feel like, kind of describe to me why someone might want to use the Terra environment to navigate through these results and run their own workflows rather than just going to the KC where they can view everything in an interactive, sort of nice packaged way? What's the benefit and utility of this mechanism?
As opposed to doing what? You mean go to the knowledge portal?
Yeah, like just seeing it through the website versus here getting all our results through the Workspace.
Oh, I mean, on the knowledge portal, you can't run your own workflows, right?
Right.
Yes. So on the Terra workspace, you can run these two workflows. One to filter the data we have by the genes that you're interested in, and the other one to run Pigeon on your list of genes.
So that's really
Which of
exciting.
course,
Where you say
which of course,
anybody can bring their own gene set in, run Pigeon, get results, and then compare them even to the results on the knowledge portal if they have a hypothesis.
I guess I'll add on another really unique utility of Terra is that it's a cloud-based workspace that also lets you connect to all of these other external resources, right? So within the Knowledge Center, we're sort of working just within CFDE space. But the beauty of Terra is that it actually has a lot of other resources outside of CFDE as well. So if you want to connect to some of those data sets or see how those results compared to what we've generated here in the Knowledge Center, I think that's a unique offering. I guess I just wanted to add a few more things. Thank you so much, Oliver. That was fantastic.
thanks so welcome.
So if you're like me and you're new to Terra, right? You've never used it before and you're not lucky enough to have someone like Oliver on your team who's an expert in it. We are fortunate to have with us another panelist who's going to be presenting at the end of the session. So please stick around to listen to her. This is Tiffany Graziano. She's a representative from Terra who's here to give us sort of a Terra 101 so that you can learn more about what the capabilities of Terra are and how you can navigate through it. One other thing that I wanted to emphasize is that this workspace is evolving. So we have documented what we've done within the workspace. If you go to the dashboard page, you'll see just sort of a list of text with some schematics. And this explains a little bit more about what each of these analyses that Oliver just described are, how you can get to them, and how they sync up to what we have presented so far on the Knowledge Center. Importantly, this is also an evolving resource. So some of our future directions are to add a Jupyter notebook which will enable you to do the same sort of CFDE GeneSet and Pigeon workflow that Oliver just described in a different format. And we're also working with our colleagues at UCSD, Kyle Galton and Peril Kadutar, so that users can actually bring their own transcriptomic data and analyze it in the same way as it was analyzed for the differential expression analyses. So we're excited to bring those new workflows and functionalities in soon too.
Hi everyone, welcome back from the break. As we head back from our initial portion, we'll be in the second half of our workshop today. So thank you all so far for participating. The next session that we're going to have is going to feature the DRC. But before we get into that, Noël is going to do a little brief recap of what we've already talked about in the first session and set the tone for the second. So I'll hand it off to you, Noël.
Thanks, Mackenzie. Thanks everyone for sticking in. Today, this afternoon, we're going to switch to slightly different topics, really focusing on the centers. You're going to hear from 3 centers coming up, and then we will also have the hands-on sort of 101 workshop with Tiffany for Terra, which is a totally different, environment for accessing data and results. So the reason we're focusing on the centers, some of these centers are very new, only a few months in. And so we're really excited to have them join us later on in the session. But Avi Mayan and his team have been through with the CFD before it started, because Avi was part of Links and many other DCCs as this whole ecosystem was getting going. So Avi has an incredible perspective and also an incredible resource that sort of spans all of the DCCs within the Common Fund Data ecosystem. And you're going to hear a little bit about that from Avi. And then Giacomo is going to talk a little bit about the tool that we referenced earlier with Jason's presentation, GeneSetCart, which is where they actually aggregate all of those gene sets that we spoke about in our earlier presentation, and how they did that and what else you can do with them. And also, so hopefully they can speculate on what we might do in the future, which is to aggregate more across CFD, because CFD is adding new centers all along and adding new data coordinating centers, and working with other resources outside as members of consortium to bring in results and data. So I'm hoping we'll hear some about that. And again, same thing, same drill. Please pop your questions. We like questions. They help us, think about what we're doing into the chat. And with that, I'll pass it over to Avi. Thanks, Avi, for being here.
Yeah, thank you so much for the opportunity to present today and to tell you a little bit about the Data Resource Center, which I'm the PI together with Shankar Subramaniam. Today I'm going to talk about this product that we are tasked to develop, and it is called CFDE Workbench. And it is a web portal for the CFDE to host data and also information. So this URL can take you to the portal, and we are tasked to create an information portal and a data portal. So you can toggle between the 2 websites, the information portal website and the data portal, and find information about the CFDE and the data sets. And today I'm really going to focus on the data portal. So our vision for the data portal for the CFDE is to bring in metadata, process data, and other types of assets from the different participating DCCs or common fund programs. So we can have a way to build tools and then form hypotheses that combine data from across the DCCs, or across from combining those data sets together. And this is really the vision of the CFDE. So this workflow show you how it works. So first we have this system where people can upload their data, and then we ingest those data sets, we assess their fairness, and we display them in this data matrix for people to upload. And then once we have all this data, which is process data, metadata, we are able to build a search engine for people to find those assets, because they start to accumulate and there's a lot of them, as well as build tools that take that data and generate those hypotheses and visualizations And this is the presentation that Giacomo is going to give after I'm done. So the types of data that we have from the different centers, and some of it is their contribution. They created, particularly the metadata and the knowledge graph assertions. They create those and upload to this submission system that we developed. And there are some other types of data sets that you probably heard of before, which is the gene sets that are organized from those different sources, and those are called XMT, as well as knowledge graph assertions. And we are also starting to gear up to have attribute tables, which are really useful for machine learning and AI type of applications. And so we divide those assets to metadata, data on the left, and also code assets. So in the code assets, we have ETL scripts that track how the raw data is converted to those processed data formats. We also collect APIs from those DCCs, from those repositories that serve this data, as well as application URLs, landing pages, and we're also experimenting with chatbot, which we're going to discuss. So once you upload your data into this submission system, the assets that you uploaded are listed and automatically getting assessed for fairness. And this is done using the tool called FairShake. And FairShake is displaying the assessment of fairness as an insignia, which is a bunch of boxes with different colors. So if it's blue, it means it meets the fair assessment answer 100%. And if it's red, it is missing some aspect of fair that the uploader may want to fix. And this is only visible to the uploaders and the submitters, the approvers from the different centers. And then once it is approved, it gets displayed in this data matrix that lists all of those assets that I mentioned, the code assets and the data and metadata assets. This is just another view. So all of the centers have their row in this table, and gradually that table gets filled up. So once we have the data, all of this data is served in a search engine. This is a screenshot from the C2M2 crosscut metadata search, where the metadata from the centers is organized in this common metadata model to identify common metadata elements from across the DCCs. And then once we have that, we can start doing a search. For example, here I search for the word liver, and I got almost 30,000 entries that correspond to files from the different data coordination centers that people can access and download. We also can search the word liver for those additional processed data sets. And here you can get gene sets, knowledge graph assertions, and also we have those gene pages that this particular gene is known to be in the liver. So the word liver came up in part of the gene's description. And if you click on it, you get to this gene page that links you out to those tools that can give you more information about the gene. So far we have about 10 of those apps and tools that sit on top of the data. And one of them is Pigeon that you heard about before, and also some other apps that we've developed. And also those apps enable use cases. So we also listing use cases that showcase how you can use those apps to perform various analysis. And one of those exciting apps is GeneSetCart that Giacomo is going to talk about. So I'm now going to pass the mic to Giacomo to tell you in detail about what is GeneSetCart and how it fits in this framework.
Great.
First, before we go in, questions for Avi? Hey, Abby, I have a quick question for you. Since you, as a data resource center, you guys are really the hub of where all the data and metadata is across the entire ecosystem. What's your sort of process, just for the audience, in terms of updating data as it comes in from the different DCCs? And how frequently does that happen?
So there is a window of data submission every quarter where we have 2 weeks open submission period where the DCCs are required to submit to us their C2M2 file, as well as encouraged to submit those other assets. But we are also constantly updating it ourselves. So we are going to the portals and we process data, and anytime there is a publication, then we are in a process of converting it to those processed formats. So this is a joint effort by the DCCs and independently by us. But it's a good question.
That's great. Any other questions for Avi? All right. Thanks, Avi. Actually, one other point. I don't know if Giacomo's going to mention this, but Avi, I'm hoping you can stay on the line for the Q&A after Giacomo's, which is one of the things that, we mentioned earlier in the session is our thoughts about beyond, the C2M2 model. These gene sets are incredibly useful resources. And we've been talking with you and others about how we would make that a more living entity as we update gene sets coming from the DCCs, but also thinking about inviting others from other like-minded NIH resources to contribute their own gene sets or participate. Do you think we could comment on that now, or should we wait till after Giacomo's talk? What will be best?
Yeah, I think we should wait for Giacomo's talk, but I'm here and I'll be able to answer questions via chat, and we can stay also afterwards and then answer all questions, and we can have that discussion.
Beautiful. All right.
All right. So hello everyone. My name is Giacomo Marino. I'm a bioinformatics software engineer in the Mine Lab, and I'm a member of the DRC team. I'll be talking about GeneSetCart, which is which you can use for obtaining, analyzing, and visualizing CFDE gene sets. So just to begin with an overview, GeneSetCart starts with assembling gene sets from many different sources. So one of those is these Common Fund gene sets that we've processed from the raw data of various Common Fund programs. But you can also actually upload your own gene sets as sort of a single gene set, or as a GMT, or a gene matrix transpose, where there are terms and then the genes contained within them on each row of the file. And then we also have other ways to actually query and add gene sets. So through Enricher, which has thousands of annotated gene sets, from gene co-mentions through GeneRiff and AutoRiff, through a resource GeneShot, as well as gene sets that we've extracted from PubMed Central articles, as well as GeoStudies using an associated Chrome extension. So we have sort of a variety of ways you can actually assemble these gene sets. And on the site, these are all assembled into a cart that is associated with the session when you're on the site. You can save these into your account. You can make them public and private. You can share them with other users through a persistent URL. And then you can perform a series of data analysis operations on them, which includes augmentation of those gene sets, combination and intersection with different set operations, visualizing them, as well as then analyzing them with many external gene set analysis tools. So this is what the GeneSetCart web interface currently looks like. So you'll see in the top left, you have a shopping cart, and this contains all of your assembled gene sets in your current session. And then you can see the various aspects of our data analysis pipeline listed here on this homepage. So here we're going to be focusing on these gene sets created from data from the Common Fund programs. So we have gene sets from Metabolomics Workbench, Glygen, Lynx, IDG, IMPC, GTEx, MoTrPAC, and HubMath. And users on the GeneSetCart site can search these by keyword or term, And then we'll highlight the DCCs which have gene sets pertaining to that term above here. So here we're looking for gene sets associated or mentioning lung. And we have some of these aging signatures computed from the GTEx data, where we're comparing expression in the lung from 20 to 29-year-olds versus 70 to 79-year-olds and looking at the upregulated genes in that comparison. So looking at these genes that are associated with aging in the lung. So if you actually select these gene sets or any gene sets of interest and press add to cart, those will get added into that little shopping cart that I showed before on the last page. So as a sort of use case that we'll examine in the presentation, we'll assemble a few of these age-related gene sets across different tissues. So we have that lung gene set that we just saw, as well as one in muscle and in heart. And we can ask some questions about these gene sets, such as can we identify genes that are upregulated in aging across multiple tissues? Can we see if there's genes known to be associated with aging in the literature related to those in these gene sets? And then can we combine this knowledge with other common fund programs such as LINKS to identify CRISPR knockouts that might mimic or reverse the expression of these aging-related genes that we identify? So in the combination aspect of the GeneSetCart data analysis pipeline, you can combine your sets in various ways using different set operations. So here we're selecting these three aging sets that we assembled, and we'll use the consensus operation to select genes that are appearing in a minimum of 2 sets in this case. So you can select your consensus criteria to find genes in any minimum number of sets that you've selected on the left here. So here we can find, if we're using this consensus of 2, so genes appearing in at least 2 sets, we find 60 genes that are shared across these sets. And we can actually go and add those back to our cart for further analysis. Another feature that we have on the site is this ability to cross gene sets created from Common Fund programs. So you can actually directly cross GMTs from the Common Fund program, such as here crossing the CRISPR knockout signatures with GTEx gene expression profiles to find significant and surprising overlaps that may help in generating hypotheses. But you can also actually cross sets from your current session or that are in your current cart. So if we select this cross session sets button here, we can actually see we have the interface to select any of these gene sets we've assembled and cross them with any one of our Common Fund GMTs contained within the site. So crossing our consensus set here containing 60 genes that are upregulated in aging across these 3 tissues, we find several significant overlaps with the LINKS L1000 CRISPR knockout consensus signatures. So we see 4 genes here appearing to be highly significant in the overlap. We see here, we're looking at the gene, the number of mentions that gene has in PubMed, and then also the number of co-mentions that gene has with aging. So overall, these genes are not super well studied. And many of these publications are actually just mentioning this gene with other genes and not actually focusing on them. So potentially some interesting novel genes to look at in the context of aging. We can also look directly at the overlapping genes that were identified between these 2 sets by clicking on this linked overlap button here, which will pop up a little modal that has the genes, the name of the overlap, and then you can actually copy that to your clipboard, send it to an enricher for enrichment analysis, or then also add it back to your cart for further analysis. So we'll actually add these 4 most significant CRISPR knockout and consensus aging gene overlaps to our cart to analyze further. So now we'll jump over to the visualization section of our data analysis pipeline. So this is on GeneSetCart and enables users to create and customize publication-ready figures that visualize the overlap of selected gene sets that you have in your cart. So you can produce figures that can be downloaded as PNGs or SVGs, as well as shared through a persistent URL. And we currently have 5 visualization options, which include Venn diagrams, superven, upset plots, cluster maps, and UMAP. So here on the screen now, we're looking at a superven of 4 of those sets with the overlapping with the CRISPR knockouts that we just saw. And these visualizations are actually interactive, such that you can select intersections here to identify the genes where they're overlapping within your selected set. So we have the legend of our different knockouts here that is appearing in each row of this superevent. And we can see that we have one gene contained in all 4 sets, and then 2 genes that are contained in 3 of the sets. And we can actually add those by clicking on these various intersections to our section over here, where we can identify HPD, HPB, and call 11A1 as our 3 overlapping genes. So HPD is appearing in all 4 of those overlaps, and HPB and call 11A1 are appearing in 3 out of 4 of them. And if we look in the literature, these genes are not directly associated with aging, but we do see some interesting trends in the literature. So HPB and HPD are both part of the beta globulin gene cluster located on chromosome 11. And they're not directly associated with aging. However, they are involved in hemoglobin production, which has been used as a biomarker of human aging. So you can see in this review paper here on the bottom of the slide. It's also possible that these genes are playing a different role besides forming the hemoglobin, or there's a potential depletion of red blood cell precursors. For COL11A1, this gene primarily encodes the pro-alpha chain, which is a component of type 11 collagen. This is also not directly related with aging in the literature. However, we see that it's associated with cancer and has been identified as a biomarker and a potential target. And it's additionally been shown to be involved in regulating cellular senescence in cartilage. So 2 aging-related processes that cancer occurring higher ages and also senescence being more directly related to aging. So potential sort of novel genes to look at in the context of aging based on this analysis. Another section of the data analysis portion of GeneSetCart allows you to send any of your assembled gene sets to one of 11 downstream gene set analysis tools to further gather information about them. So here we're looking at this consensus gene set that we assembled from those 3 aging-related signatures across the tissues, and we're sending that to CHIA3, which is a transcription factor enrichment analysis app, which uses a variety of transcription factor target databases to predict the TFs that are most likely to be regulating your gene set. And here we see KLF1 as the top ranked transcription factor on the top of this bar chart. And that's actually really nice to see because KLF1 has been directly linked in mice to promoting healthy longevity and delaying the onset of age-related decline. And the last sort of section of the GeneSetCart app is this ability to produce a report. So this enables users to select up to 5 sets, choose their specific visualization and analysis tools, and then produce a comprehensive report about those selected sets. So this can then be organized by the gene set or by the analysis type. It can be shared with the persistent link and can be exported to a PDF. So here on the right, we see an example of the top of this report where we have a link, persistent URL to the report, a way to dynamically organize by gene set or by analysis type. For instance, if you want to compare the analysis of 2 of your selected gene sets more easily, as well as a button to download the report. We have a persistent link to the GeneSetCart session, the selected sets, an automatically generated abstract based on your chosen visualization and enrichment analysis tool options, and then a hyperlink table to quickly navigate to different results of interest. Importantly, we also have a hypothesis generation button here under the enrichment tools analysis option. And this enables you to generate a hypothesis of why 2 sets might be overlapping when you select 2 sets from the set selection menu above. So we do this through using an LLM, in this case GPT-4o, where we provide it along with the system prompt, gene set, the terms of each gene set, the overlapping genes, as well as the enriched terms from several libraries, which includes Go Biological Processes, Wiki Pathways, MGI mammalian phenotypes, and the GWAS catalog, asking the model to produce a reasonable explanation for the overlap using those enrichment terms and the various information we've provided it. So here we're looking at one of these knockouts, the DSTYK, along with our consensus aging set. And we see that we actually have these various enrichment terms baked in to our hypothesis, where we can hover over them and see the specific rank and enrichment values and significance of that term using those overlapping genes. We see we have some blood-related terms here, anticoagulation, abnormal iron level. And so it's going to produce a nice hypothesis, which can give you a reasonable explanation for why you might be seeing an overlap between these two gene sets. And this functionality, importantly, is also available when crossing CFDE gene sets or GMTs on the crossing page that I showed earlier in the presentation. So just in summary, GeneSetCart is a user-friendly platform that enables researchers to analyze and manage collections of gene sets. This can be used to compare gene sets generated from 8 NIH Common Fund programs currently. You can also utilize an LLM to generate hypotheses for pairs of gene sets that have high overlap and are potentially unrelated. We've also shown how you can combine the GTEx and links data to identify aging-related genes, reveal mechanisms, enrich transcription factors, and identify potential targets. And there's also many other use cases actually on the site if you want to check out how you could also use this platform in different ways to facilitate discovery. So in the future, we'd love to be updating the site with more Common Fund GMTs as we have them. With that, I'd just like to acknowledge everyone in the Mayan lab for their work on this site, and especially our former lab member, Tiffany, who developed much of this site originally before going to pursue her master's. So thank you for listening. Happy to take any questions.
Thanks, Giacomo. That was fantastic. We had a question earlier during Jason's talk, which I think might apply to some of what you've covered here about co-expression and how users can integrate co-expression data. Is that functionality available here?
It is indeed, yes. So the augment portion of the data analysis section on GeneSetCart, which I didn't show in this presentation, provides 3 ways that you can augment your gene set. So one is with co-expression sourced from ArchIS4, which has uniformly aligned samples from the gene expression omnibus. So we use that to create correlation matrices, and then you can augment your gene set with co-expression. And we also have protein-protein interactions that you can also augment your gene set with, or with literature co-mentions. So 3 mechanisms there.
Thank you.
Any other questions in the chat?
I have one if there's time, Mackenzie. I'm curious how we're doing on time.
Absolutely. We still have 4 minutes, so
Right.
feel free.
Beautiful. I have a question. Maybe it's for Avi and Giacomo, and maybe it's also towards us as the Knowledge Center too. And I don't know if Jason's still on, but earlier we talked about the linking of these gene sets that you all have aggregated to human phenotype. And now if you're on the KC, you can actually say you could search your favorite phenotype and find some gene sets that may be associated with it. But the challenge there is if you actually find something you want to follow up, you need to know like the provenance and the domain and what that gene set was curated to measure. I know that the DCCs have done some work to this, and you've done some work, Avi. Can you talk about how we might do a better job as an ecosystem of curating those gene sets as people are making, assumptions and hypotheses and using them, so that they can trace back to what they are, or at least contact the DCC as a first approximation? But I'm curious your thoughts on how we might best do that.
Yeah, I think it's a good idea to have, the scripts that were used to take the data and convert it into the gene sets. Those should be very visible and accessible. And then in some cases, the DCCs are, they can guide, and people can go and dig deeper to get more information than the gene set. So one of the limitations of those gene sets that we basically abstract the data for data integration, but we throw away, for example, the ranks and the order of the gene set and the values that come with them, or the p-values. So different gene sets come from different sources, and if it is expression, the differential expression level matter. If it is from genomics, whether it's the type of variant it is, and how close it is to the gene, and whether it is whatever causing deletion, or those details are thrown away when we're dealing with gene set. So that's why moving forward, we also want to move towards like attribute tables and ranked sets. But the provenance is not a difficult issue to solve. So first you want to link the gene sets to ontologies and dictionaries, so their description is more detailed. Right now, we're not doing very good with that. And also the scripts, then those can be provided as uniformly formatted Jupyter notebooks with making them open and linkable through GitHub, so people can also update them easily.
And I just add on the ETL section of that data matrix, there are many of those scripts used to create the gene sets. So doing maybe a better job of linking, from GeneSetCart or from the KC to those scripts so people can more easily access and find how they were created.
Brilliant. Excellent, because I think that's what we're really hoping to do across the KC and the DRC is to help navigate that. So I just want to give people a taste of what we're thinking about. Perfect. Other questions for Avi and Giacomo? All right. Thank you both.
All right, everyone. We're going to head into two other center presentations as we move on in this session. So first off, we're going to hear from the new training center, and then we will follow with the cloud computing. We just wanted to give them the opportunity to introduce themselves, share some of their goals, and please feel free to use this as a time to get to know them. So I'm going to hand this off to Jennifer and Allissa. So thank you.
Thanks, Mackenzie. Let me screen share really quickly. Okay, can you see my screen?
It popped off for a second, but it
did
did
that
work.
last time too.
Yeah.
So your
One
video is
second.
working. That's
Good.
for troubleshooting. Oh,
Let's
this
try
is
this again. Better?
Looks
yeah,
perfect. Thank you.
Wonderful. Okay, I had to move it to a different screen, so I'll be awkwardly looking away. But hi, I'm Jennifer Burnette. I'm the project director for the CFDE Training Center. We are one of the newer centers, so we started in around September 2024, so fall of last year. I work for ORU, or Oak Ridge Associated Universities, and we partnered with Biodata Sage for this training center. Oh, no, I can't get it to go to the next slide. Give me a second, guys. Uh-oh. I may have to restart. Hold on one second.
That's okay.
Can you all hear me? I think I just froze up. Allissa,
Yes,
you
I
may
can
have,
hear you.
you may have to pull them up for some reason. My screen just froze up.
Usually that means your computer's about to crash, so.
Oh, good. Let's hope not. Okay, hold on one second. Let's try one more time here. Okay. Awesome. All right. So getting into the training center goals, you've heard a lot about the different CFD partnerships that go on across the centers and all the funded programs like the DCCs. And one of the goals of the training center is to coordinate with all of those partners to meet the training needs that you all as the end users have. Something else that we look to do is support directly current and potential learners that are engaging with CFD data, and ultimately expand the CFD data user base. And with that, we want to enhance confidence and complexity of the data set usage. So we're starting to get some of our branding in place for the center, and we've landed on a tagline of decoding the data ecosystem. So even though we've only been stood up for a few months now, we are currently either actively producing some training things or very close to launching. So we're going to talk about several of those today. So the first one I want to talk about is the mentoring program pilot. I want to emphasize the planning is in process with this one. However, what we're looking to establish is a 10-week virtual program during the summer of 2025, where we're going to pilot essentially engaging mentee teams, a faculty PI, and a student at a university to partner with a mentor at one of the data coordinating centers. or another mentor that is working closely with CFD data. So we're really looking to emphasize that this is likely a mentee team that's probably already working together. We do look to offer a stipend for the team that's going to participate, but it's not going to be something that would fund your entire summer, for instance. So it's really kind of an extra supplement and an interesting way to engage with some new data that you maybe would not have otherwise. So if you're interested or want more information about it, we are going to start recruiting for our mentee teams in February, March of this year. So just in a couple of weeks, couple of months for those mentee teams. So if you're interested, we'd love to have an email from you. The other thing I want to talk about is our podcast. It is called Decoding the Data Ecosystem. So we just leaned into our tagline. Noël was our first guest on the podcast. So that was great to have the Knowledge Center represented in our very first one. The goal with the podcast is really just to give a venue for a much more casual conversation about omics research and deep diving into some of those things that you may not get into if you're taking a training or keeping things more high level. And Dr. Allissa Dillman, who's on here with us today, is the host of the podcast. So she is going to be the one that you'll hear in all the podcast episodes, and then we'll have rotating guests talking about a lot of different omics related topics. So with that, I'm going to turn it over to Allissa to talk a little bit more about our other opportunities and things that we're doing. Allissa?
So we are also working on a monthly how-to seminar. Right now, they are currently on Vimeo. We have two that are currently released, genomics and transcriptomics. The idea behind sort of these first year of how-tos is to really build a foundational knowledge on these different omics types, and then to point to where you can work with these omics types in CFDE. So kind of connecting sort of the biology, the technology, and the data sets. Once we've got a good foundational set of how-tos that kind of guide through some of the basics of genomics and transcriptomics, spatial and single cell, epigenetics, proteomics, and things like that, we will definitely be working with the Quick when they have their cloud platform up to do more technical deep dives into sort of more hands-on how-tos, with the idea that we can then look back or reference back to those foundational how-tos to sort of build a nice training path. Yes, and like I said, we're on Vimeo, so if you want to check that out, you can go to Vimeo and do CFDE Training Center. Next slide. However, we do want to create a much more interactive experience with all of the materials that we're generating, but also potentially things that other CFDE DCCs are generating in terms of training. So we're actually working on a learner dashboard, which will have a wide array of training modules. And the idea is that we're going to use this platform as a way to engage with both beginning users, because there'll be a lot of interactive features, quizzes, knowledge checks, hands-on exercises, and things that are very interactive. But we can also point to much more complex training modules, such as what might end up in the Quick. So that way that we can sort of help learners again with that learning path. And things will be tagged based on their complexity, based on their omics focus, and other things like that, with the idea that this is going to be sort of a landing space for all things training, so they don't have to kind of pop into all different places. This is still a very, very much a work in progress, but we're hopeful that we'll have this out in spring of this year. Next slide. If you want to engage with us sort of in a much more creative way, where you get to bring your expertise and resources and data to the table, we're actually going to be at the BioIT World Conference. We're doing a fair hackathon, and the idea is, we're really interested in bringing CFDE data sets and tools and things like that, but also having you all bringing in your data and tools that we can sort of integrate and interoperate on, and see how all of that works together in a fair ecosystem. So if you have ideas about a project, you can pitch it. at the website link there. However, if you're a little more interested in just participating in the next slide, you can also just join a team. So if you're really interested in just seeing and learning more and having real, true hands-on, activities, there'll be a whole bunch of different kinds of projects that we will have available that you can participate in. We're really going to have partners all throughout CFDE and all of you and technical people and scientific people, so you can really kind of build interesting things and test things out. But yeah, we'll be that the hackathon again is in BioIT World in Boston, April 1st and 2nd. Next slide. But yeah, that's the training center. Please reach out if you have any questions about any of the things we've presented. Thank you so much for listening.
Thank you. There's a question in the chat asking if you could put the link to the podcast in there. I think it would be great for people to take a look at that.
Thank you. I'll drop all the links in the chat. I couldn't see it with the screen share.
No worries.
Yeah, and as I said in the chat, if you've published with any CFDE data, we would love to have you come on to the podcast and we can chat about your really cool research and hopefully get that out to even more folks to go check it out.
Awesome.
Thanks for having us.
I have a question. We have time, Mackenzie?
Absolutely.
So first of all, I enjoyed being part of the podcast. Thank you, Allissa, for inviting me, and Jennifer. That was fun. One of the things that, CFD as an entity you just started to do is really make a social presence vis-a-vis with this workshop and going to webinars and conferences and really getting out there. I'm curious as to, what do you guys think as the training center, really being at the thick of some of that work, trying to get people to be aware of the CFD. I think that's what events like this will do, but also exactly as you suggested with these hackathons, what do you think is the greatest challenge in a resource where there's 18 data coordinating centers and 5 centers for you guys in trying to sort of synthesize the ecosystem to a broader research community? And is there anything you can draw on from your other experience in, other collaborations that might be valuable here? Just give us some insights, because I think a lot of us are thinking about it with you.
Allissa, you want to take that one?
Sure. I do think that, there is such a wealth of tools and knowledge and trainings, and they are very difficult for learners to find across these sort of different places and centers. Some of the interviews that we've had so far, researchers have really expressed that, more often than not, they're still finding this data because of a publication first, right? They see a really cool publication, and then they slowly backtrack, and then they eventually find data, and then they eventually find that data is part of the, this whole ecosystem. But I think, especially for beginners, it's still really hard to sort of see, okay, I have my niche piece of research here, and I found my niche data set, but I still may not be really clear on how that connects to other things I could pull in. So that's why, like I said, we're really focused on trying to create that foundational understanding of are you interested in this overarching topic? There are actually a couple of different places that you can look for this data. And hopefully starting to, when we do our hands-on activities that are on the more technical side, sort of pulling in multiple data sets and sort of helping people understand why that's important. And the same is true for the hackathons, right? There are a lot of people that'll come in knowing a lot about omics, but not necessarily knowing a lot about CFDE. And seeing what kinds of tools and what kinds of data and where they're going will help us sort of make sure our communications are also going to where they are.
Beautiful. Jennifer, any comments from your end?
No, I was too busy putting links in the chat.
Even better.
Thank you.
Already starting the training. I love it. All right, beautiful. And I think, as the centers from particularly the KC and I believe the DRC's perspective, as centers responsible for kind of aggregating knowledge and data across the ecosystem, I think we're excited to have the training centers part of our ecosystem, because we can really lean on them to help, create educational material to help make this more useful to other people, because they do that well. So really happy to have them on board. Any other questions for Jennifer or Allissa? All right, Mackenzie, want to introduce our next speaker?
We will now have John Fonner, who will be introducing the Cloud Computing Center for CFDE. So with that, I will hand this off to John.
Great, thank you so much. And let me go ahead and pull up sharing. Please let me know if this comes through correctly.
It looks great.
Okay, great. I'm really excited to be here, happy to be here, happy to be on the heels of the training center's presentation as well. We're really excited to work with them. We are the other new center with CFDE that started in the fall. We are just bringing up services now, but I'm excited to tell you what we have planned and what we've been working on. I'm from the Texas Advanced Computing Center. I'm leading the QUIC team, as we are called, the Cloud Workspace Integration Center, but I'll just call what we do the Cloud Workspace. I know there are so many acronyms already. Okay, so what is the Cloud Workspace, and what is it that we are trying to do? First off, we are connecting this workspace to CFDE data. So this will be one where we will integrate with both the Data Resource Center as well as other DCCs to be able to import data into this workspace very flexibly. So as you're wanting to combine data sets, as you're wanting to bring them alongside your own data sets, this will be a great space. We're bringing in a very extensive app catalog and workflow set that has been in the life science computing space for over a decade. So hopefully people have lots of tools right out of the gate. The computing infrastructure that will be backing this cloud workspace is very extensive. It's being anchored by the academic cloud at the Texas Advanced Computing Center with its high performance computing resources and cloud resources and data storage and all the other nodes that are there, as well as extending to the commercial cloud for those that would prefer to or need to be in the commercial cloud. And we really have support for all academic researchers. We want this to be a place where people can come and collaborate with people across all institutions. We want this to be a meritocracy of good ideas that are able to compute against this data to make discoveries. And so for institutions that may not have their institutional resources to do computing at scale, we want this to be a valuable resource for you alongside with everyone else. All right, so the QUIC team. I mentioned the Texas Advanced Computing Center. That's where I'm from. I've been there for 13 years, and in this picture, which we just took recently at a team meeting, I realized that 13 years is not a long time compared to the experience that so many of these people bring. Almost everyone in the photo has been working on supporting bioinformatics and life sciences computing for academia for over a decade. We have partnered with Penn State University. Anton Nekrotenko is leading that part of the project. From Johns Hopkins University, we have the Michael Schatz Lab. And from the San Diego Supercomputing Center, Shava Smolin is representing the CloudBank project. And it's just a really fun team to work with. These are all people who are passionate about bringing research, out into the world and making it easy and available to do that and to publish and to make discoveries. So what is the problem that we're trying to solve? Well, we want to provide accessible, reproducible analysis at all scales for all researchers. I think that sums it up pretty tightly. By accessible, we want to be able to provide interfaces that are familiar, whatever has the greatest utility for the person who is working there. We want to have software that's already available that people don't have to figure out how to install, and support really to be able to access all of these things from a staff that has done this with their own work and that has helped many others, experienced staff. Reproducible, a lot of times work is done on someone's local laptop or in some way where it can be difficult for someone else to reproduce it. The platform, the Galaxy platform that we're building on, and the work that TAC has done has always had a strong emphasis on reproducibility. We want to see people making discoveries and then putting in links in their publications so that the editors and reviewers and readers can go back and with one click reproduce that work, and then start using their own data and extending what was done to make new discoveries. At all scales, there's no, we work in high performance computing, so a lot of times the features things that are very large scale, but the average job size at TAC and the average job size through Galaxy is just one node, and it's just one task that's being run. We're very aware that in the bioinformatics workspace, there's a lot of small steps that we need to perform in our workflows to be able to transform data types and to be able to do these analyses. And so we really are targeting things at all scales. Nothing is too small and nothing is too large. And then finally, we want to, again, be available for all institutions. It's very permissive on creating an account in the platform and being able to get started. And this cloud workspace is backed by a fair amount of resources right off the gate. So we don't need to worry about getting your credit card information to get you going on the platform. We're a nonprofit center. As part of this project, we've already included computing and data storage resources to be able to get people going right out of the gate without any barriers. So for tools and interfaces, we've already seen some great tools this morning. We want to complement those. We have the Galaxy interface as the kind of front end of the portal. These screenshots are just examples. We have not, I don't have a link for you yet. You'll see this in the end. I'll talk about timing in the end. But you know, Galaxy has served over 500,000 users now. It's something like over 540,000 users that have made accounts just on the main instances of Galaxy in the US and in Europe. And so there are lots of familiarity, lots of testing that has gone into that interface. The app catalog that comes alongside that and the interface that's familiar to folks will be kind of the front door to working on the cloud workspace. But additionally, we want to provide interactive environments, command line environments. If you're coming in and saying, I already know what I want to do, I already have scripts for this, you know, I'm already well versed in Python, or have a high degree of automation that I've built up, we want you to be able to bring all of that sophistication into the cloud workspace and operate in a way that you're comfortable. We want to provide resources and then otherwise get out of the way to let you be productive. When it comes to bringing in data, which is always the second thing to do work anywhere, you need your apps, you need your data in place. For data, we'll be combining everything that we can. So like I said earlier, we've been working with the Data Resource Center and other data providers to integrate with their data sources. There will be ways to export data from the DRC and import it into the cloud workspace. That'll all do that on your behalf so that you can work on it. And then combine it with your own data that you can upload. And also combine it with other external resources that we can connect to, so that hopefully there can be a lot of innovation on the data side and not a lot of difficulty in getting it there. The resources we're providing are connected to the Internet 2 backbone, the academic national research network. And so it's a great place to be able to access data, even at volume, to come in, to do work, to be able to share that with others without having to download and upload and worry about how much storage space you have. And then as far as computing, we have a lot of CPUs in the data center, all different types, AMD, Intel, ARM architecture, x86 architecture. We have lots of GPUs at the center for apps that can take advantage of GPU computing, for AI-related workloads. Those are there. We have data storage at scale, specialized node types that can handle problems that need large memory in order to be able to get all the data on one node to do that type of work. And then really our staff that is at TAC and the staff at the other centers as well that's supporting this project. There are very few computer scientists among us. We are all domain researchers that have done this type of work. And so if people have very specific questions about a tool or some struggle with installing something, if they need to install something or whatever it may be, you'll have people that are ready to help in a very domain specific way. We're not just providing a platform and then telling everybody this is where our responsibility ends. We're able to really dive in and help you be productive. We've done all types of computing at TAC. In the life sciences space, we have calculations in femtoseconds and angstroms, and then in the astrophysics space, it's gigayears and megaparsecs that are the units of measurement. So we want to be this comprehensive environment for people. Provisionally, our timeline, this quarter, we are opening up the cloud workspace for internal users. We're doing our own development and testing. We're working with our partners to integrate workflows and documentation and training and data. Q2 of 2025 will be our initial public deployment. That deployment is focused on open science data, so not regulated, protected data that needs that extra security layer. We'll support private user data. We'll support CFDE data. And again, working with our partners, this will be a constantly expanding set of data that you can bring in from different sources. By Q4 of 2026, we have targeted support for regulated data. This is all the protected FSMA, dbGaP stuff that we also want to be able to support on the platform. Again, this is provisional timeline. We would love to beat these deadlines. Hopefully we don't slip beyond them, but this is definitely our plan. So right now the portal isn't live. Catch me in a month and I'll have a link for you. If you want to reach out to us, and why you might want to reach out to us if you're interested in this tool so that we can target your use case early on, we would love to know about it and help you use the site and be integrated and be able to use it very early on. You know, if you have data that you want to make available on the site, all those types of things, then please reach out. A couple of addresses you can use. One is CFDE at consult.pac.utexas.edu. This will go to the whole team and be available. There's my personal email address on there as well if you want to just reach out to me. And once the portal is live, you'll find a link on the CFDE info page. I don't know if this has been talked about before, but links to all the centers are available on that page. So with that, I'd like to just thank the NIH and the Common Fund for the opportunity to be part of this project. You know, this type of work is what we most love to do. And so to have the opportunity to do this for the CFDE community is really wonderful. All right, thank you.
Thank you. Do we have any questions from the audience?
John, I'm curious about the fact that just because I'm not as familiar with cloud infrastructures as I'm sure you are, and I'm really excited to see Galaxy as part of it because I think it brings another cloud provider to the ecosystem. Have you thought about how, given that some of the DCCs themselves operate in different cloud environments, like Kids First is in Convatica, and I'm sure there's some others and I'm not thinking of, we have data now in Terra. What are your thoughts about how to kind of make that, how Galaxy is going to interface with that, and how, as the cloud computing center, you want to make it clear to users where to find things? I guess that's my question.
Yeah, sure. Yeah, and just I could bring up that diagram again, but you know, at the back end, we're all using servers and computers. The things you mentioned are mostly in the commercial cloud, be it Amazon or Google. You know, the data here will be starting in the academic cloud. But a couple of things. This interface layer, be it Terra or the other centers that you mentioned, the other interfaces that you mentioned, you know, Galaxy is an alternative. I think what will set it apart is the comprehensiveness of what it's able to support. We are building this with the mind that there is no one set of tools that will encompass all of research. Everyone, by nature of what they're doing, is seeking to do something novel. And so if the novelty is coming on the side of, I have different data sources or different combinations of data, but I want to use standard tools. We want to provide those workflows as much as we can. And I think in that sense, we'll be an alternative to some places that are also providing those tools. Hopefully the comprehensive piece comes in that when you're no longer able to fit into the apps or tools that are there and want to do more data exploration on your own, we'll have interfaces and ways to go and deploy your own code and make that part of the ecosystem. Galaxy's been, you know, community driven for 15 years, and it has a whole massive community behind it that can deploy tools, or you can just use tools directly, and really a lot of leverage that you can turn there to do whatever you need to do.
Perfect.
Great.
Other questions for John? All right. I think, Mackenzie, are we at time to transition? Thanks, John.
Thanks, John.
We have to transition to our Terra 101 now. Right?
Yes. So, but did you want to take a moment for?
Yeah, I just want to make sure we have time. Tiffany, we have some time, right? Do you have the full hour, or do you kind of have some flexibility for a minute or two?
Yeah, I'm flexible. So
right. I just have like one set of slides to sort of wrap up this section, and then we'll turn it over to you, Tiffany, which would be great. I'm getting my slides up. All right, just more to thanks all for hanging in there. I know this was a longer workshop, but the beautiful thing is hopefully you got up and take breaks and just stretching. We didn't have coffee, but hopefully you had coffee at home. I'm hoping today that, you know, if you tuned in for part or all, we'll have recordings, and we'll make them accessible to the event, all the centers, but also on the KC and on the DRC for you to take a look at if you missed it. But I'm really hoping today, you have a little understanding of what the Common Fund data ecosystem is. You've learned that there are many components, but our job as we still are early in the process of, synthesizing the entire ecosystem into one entity. You've learned about some of the components, and we've hopefully enticed you to check them out. I hope you have a basic understanding. It's been really great to see some of the tools, analyses that have been conducted across the DCCs, some work that the DRC has done to really bring all of the data together into one, model that really has been sustained across the ecosystem. The C2M2 model, actually, as I've been thinking about it, I see that model in other NIH RFAs as kind of like a paradigm for how, resources of resources. So like if you have a, NIH resource that's supposed to, collaborate with another to create, some shared resource or shared knowledge or results, the C2M2 model has a very nice foundational principle. And I think it's very extendable to other resources that are NIH funded. So it's great to hear about that today. And I think that's a really cool product of CFDE that the community of CFDE has really adopted. And I'm hoping that we can extend that sort of grassroots effort to GeneSets, because I think that is a language that will even reach beyond CFDE to so many other types of consortia and research projects that are generating, molecular signatures and reference maps, and really using GeneSets as that commodity. And so we're hoping that that can be something that we advance. And that's kind of why we put everything into Terra. That's why we've been working closely with Avi's team and Giacomo to take all the GeneSets that have been generated and translate them and link them to human phenotypes, because that gives them another way of being useful to you if you're studying a trait or if you study a gene, right? So we're hoping that some of that did that for you today. Again, we really hope that you stay tuned for the next section, which really features a close collaboration with us at the Broad, the data science platform, who has Terra and has done a great job of representing other NIH resources, Anvil, ClinVar, Biodata Catalyst, and whatnot within that infrastructure. So Tiffany's going to talk you through all of that, which I hope you can stay for. But like just one last thing, one of the things that I've learned being part of this collaboration is that the resources are amazing, but the people are even better. And being part of this ecosystem has been awesome. And that's why we really started these public events, is because we wanted to talk to scientists directly and have scientists talk to you directly, because that's how the resource really gets a life of its own and is shared more broadly. So it was really great to have you all here. I hope you all join with us more. We have webinars, as Mackenzie mentioned. I think she's going to wrap up with that at the end. But if you want to just talk to us directly, we don't have a just CFD generic email that goes to everyone in CFDE, but we do have little ways of doing that, and we're starting to get that a little bit more concise. But if you're interested in contacting us, please start with this email. We will get you to the right person. It goes to many of us that are panelists on this call, many of us in CFDE, and it will get you to the right data type, the right session, the right question answered, and we're pretty good about getting back to you really fast. But more importantly, if you're interested in collaborating or working with us at all, that's actually something we care about deeply. So please talk to us via this or email us directly. So with that, I want to thank our earlier speakers, but I'm excited because, Terra does these types of workshops a lot, and they're giving us sort of a truncated version really about, how you bring CFD users to the Terra environment and bring the Terra environment to CFD users, and vice versa, sorry, you know what I mean. And then we'll be talking through some of the ways you can actually use Terra and the mechanics of it. I will stop sharing. All right, there you go. Hey, Tiffany.
All right, should I jump right in?
Yeah, that'd be excellent. Thank you.
No problem. All right, let me share my screen.
Looks great. Thank you.
Awesome. All right, everybody. Well, hello. Thank you so much for having me join today, Noël, Mackenzie, and team to talk about Terra. So my name is Tiffany Graziano. I work in the data sciences platform at the Broad Institute, and I'm responsible for the team that does a lot of these trainings. So I'm excited to present one for you today. So in this workshop, you will learn how you can organize data, tools, and resources in Terra. We'll talk about how you can create different compute environments to do batch analysis using Whittle workflows and more interactive analysis using Jupyter Notebooks, Galaxy, RStudio. And then I'll give you a brief demonstration on where you can find example resources to get started. Okay, so these first set of slides will just walk you through kind of like why we decided to build something like Terra. So Terra is a biomedical data analysis platform that's built for people to be able to access data, run analyses, and collaborate on the cloud. And I don't need to explain this to many of you. It's pretty clear that data generation is on a roll with all these different programs, creating more and more data. And our traditional approach of accessing that data by bringing data to researchers had a few issues with it that this cloud approach is trying to solve. So a few quick notes on that is that in the traditional approach, data, is copied to local storage. It's not shared. That means that everybody who's making copies of the data has to worry about storing it and paying for that storage. And everyone has their own copy, so there's no source of truth. Results are not reproducible oftentimes because they're done on an isolated environment using custom tools. John kind of alluded to that earlier in his presentation. There are high infrastructure costs, especially, as you're getting bigger and bigger data sets, you're going to have to build bigger data centers to house that data. And then also individual security implementations are not, always of the highest standard. And so people that are providing this data can be, concerned if there's copies living everywhere and there's, weak security in one area where that data is, especially for controlled access data. So the cloud approach is meant to be the solution for these problems with the traditional approach. So cloud-based data storage means you have immediate data sharing. You pay for a single storage instance that many folks can access. There are shareable tools and containers, and you can create whenever you're doing your analysis on the cloud, you're basically designing it so that it will be reproducible. Someone else can pick it up and recreate what you've done. And then it's more cost effective if you're thinking about elastic compute and how you might want to just analyze the data once, but not, have to pay for it for a long time. You just pay for the computation that you use on the cloud when you use it. So clouds are hard to use out of the box. So if you wanted to go use the Google Cloud Platform or Azure, et cetera, there are a lot of things that you have to set up and infrastructure and things that you have to worry about that you don't necessarily have to worry about anymore when you're using Terra. So Terra is trying to make that easier for you. So we'll go into our intro slides here. So Terra, as I mentioned earlier, is a biomedical data analysis platform. We have collaborated with Microsoft and Verily to build Terra, and other NIH programs have helped develop it over the years. I think they were mentioned over the call, but Anvil, Biodata Catalyst, the NCI, several different NIH programs have been involved in bringing Terra to what it is today. And so going back to the core capabilities of Terra, we have a data library where you can find controlled access and publicly available data sets, and you can bring them together in a place to analyze them. We have workflows. So the workflow language that you can use in Terra is called WDLs. It's the workflow description language, and you can run workflows at scale, and you can, use other people's workflows that they've written. For example, Oliver was sharing the workflows for grep genes and pGen, and now that those are available in a workspace, you all can use those workflows too. There's also interactive analysis, so you're able to launch applications like Jupyter Notebooks, RStudio, and Galaxy, and then interact with the data that you have in the cloud. And then finally, what brings all of these together is this concept of a workspace. And that's where you bring the data, the workflows, and the ability to launch these interactive tools all in a single place. And you can collaborate, with your lab in like a single workspace on that data. So yeah, Oliver shared the CFDE workspace that is in development. So that was an example of a workspace. Okay, so this slide just gets at that Terra isn't an island. It's a part of an open ecosystem. It's modular, standards-based, and it has open source components. This means that you can Find and access data, bring your own data into the platform. You can access tools, but you can also bring your own. So there are tools mentioned in Galaxy that are available. We're integrated with DocStore, and I'll show you that in a little bit, but DocStore is a great spot to go find WDL workflows and different notebooks and things that you might want to do reuse to analyze your data. And I think the only thing else I'll mention is you can perform analyses and share securely with our built-in security features as well. So talking about that, you can work with highly sensitive data in Terra, like human genomes and clinical data. And we have a centralized security, which means all of the components in the Terra ecosystem exist within a FISMA and a FedRAMP moderate environment that satisfy the security requirements for many controlled access data sets. So if you want to look us up, we're in the FedRAMP marketplace, and that's where you can read more about these particular security protocols. Okay, so we often get asked, what about PHI in Terra? Is that okay? I have a link here to our terms of service and security posture, so you can read more about the latest there. But basically what that boils down to is it really depends on your organization and the contractual obligations they may need around protecting data, including breach response. So usually people anonymize their data before putting it into Terra to avoid having to figure out those answers. But if that is not an option for you, you can email us to start a conversation. So we have a support team. You can reach us at support@terra.bio. And basically, we have a staff support team that's there to help you 9 to 5, business hours, Monday through Friday. So they not only can help you with questions like this, but they can help you with also your analysis and things that you're running into as you're using Terra. All right. So I'll pause here to see if there's any questions before I go into a tour. Okay. All right, so I'm going to show you all a tour, but before we jump into that tab, you can follow along with me if you'd like. Google Chrome is the browser that Terra works best on, and so if you want to go to app.terra.bio, I will be there in just a minute. But first, I wanted to again talk about the core capabilities and one central location of what you're doing in your workspace. So in your workspace, which I'll demo in a moment, you can think of doing your entire research project inside of this workspace. So you bring in your inputs. That's where you bring in your raw data and your resources, and get that going in your workspace. And then you can do some of your next steps might be processing and analysis. That's where we envision you would use your WDL workflows to clean up your data so that you can then interact and analyze it. Create visualizations or perform statistics on that data. And then finally, you can, share your data on, say, maybe you use bioRxiv and you want to share a link to your workspace. You can actually write into the support team and make your workspace public and or featured so that you can then share it with the world to go view and see your data, see your tools, see everything. So that's a pretty cool way of making your research really transparent. Okay, so if you'd like to follow along with me, I'm going to app.terra.bio. I'm going to navigate to the library and featured workspaces, and I'll show you that in a second. And then we're going to search for the COVID-19 workspace. So I'll come back to this slide in a minute to remind you, but here we are at app.terra.bio. This is the homepage, and you can see there's already some quick links. You all may have to sign in. If you might see the sign in modal. I have already signed in, so that's why I don't see it here. All right, so yeah, we have some quick links here to viewing my workspaces, viewing examples of other people's workspaces, or browsing data that we have available. I also wanted to call out this new thing that we're providing is this Terra roadmap. So basically you can see what we are planning on releasing in Terra. And you can also see previews and participate in a beta portion to preview features that we haven't released yet, which is pretty cool. So up here in the menu icon, you can see that I'm logged in here. And I have a few options below this, my profile, groups, which is like basically a concept of like Google Groups, which you can use to make it easy to share workspaces and things like that. You can share it at the group level instead of individuals. I can see more billing details here. And then I can see this concept of a cloud environment, which I'll go into in a little bit more detail later. Then I have my workspaces link, the library. So the library is again where you see that, the collection of data sets that you can look at, the featured workspaces, which are kind of like our most, defined and good examples of different types of analyses people want to share. And then finally, this link goes to workflows, which is a way to quickly access DocStore, and I'll show you that in a little bit, or the Broad Methods repository, which is within the Broad a place where people, quickly upload their WDLs and do testing and whatnot. There's also curated collections from different communities. So the GATK community, best practices, long reads, pipelines, the WDL analysis research pipelines. So we have a team in the data sciences platform that basically works on pipelines for different NIH projects, and all of the pipelines that they develop in collaboration with different scientists get posted here for open use. And then our viral genomics group. Okay, so just going back to the menu again, I wanted to show you a few more things. So for platform news, here's where you can find a link to the Terra roadmap. You can see and preview new features that we're planning on releasing and be a part of the beta. You can request a feature. You can also look at the release notes. And then finally, support. We have a separate support site with tons of documentation about everything you can do in Terra. We have a community forum, so you can search for, if you have a question and you're like, I'm sure somebody else has asked this question before. We have a very long-standing community forum, so there's a lot of good intel there where you can search for your question and might find an answer. We have the ability to kind of contact us right away. So if you have a question, you can just send us a quick note and we can get back to you there. And then I think that's everything I wanted to talk about. Oh yeah, and then just service notifications. If we have scheduled maintenance or something happens, you'll find more details there. Okay, so I think at this point, what I'd like to do is navigate to our featured workspaces section. And if I have more time at the end, I can go through some of those other bits in more detail. Okay, so here we see the different featured workspaces. So we have workspaces that are available on the Google Cloud Platform, and we have some available on Azure as well. Terra is primarily on the Google Cloud Platform, so I'm going to focus on that today. We have getting started tutorials that our user education team has developed. So lots of really good stuff in here if you are just getting started. Different organization of analysis tools, experimental strategies, technologies, scientific domain, data sets. So a lot of different ways to filter. What I'm going to do though is go to the COVID-19 workspace. Something really interesting that we learned post-pandemic was there are a lot of public health labs using Terra to do surveillance and do their public health analyses in Terra, which is really, really cool. So I'm just going to show you the COVID-19 workspace because it's got a lot of different components referenced in Terra that I'll show you. And then we can also go back to the CFDE workspace too. So first thing I'll mention is a little bit more on the infrastructure of the workspace itself. So whenever you create a workspace, it actually creates a Google bucket. And what's nice about that is you have a place then you can come bring your files that you might want to analyze in Terra so that you don't have to, then maybe they're just on your computer and you're not sure where you want to put them. That's a way for you to bring those files to the cloud really easily. That being said, when you're in your workspace, you can actually analyze data that is living in other Google buckets as long as you have permission to that Google bucket. And we have, articles on like how to make sure you can share other Google buckets with your Terra account so that you can analyze data living in a variety of places. So again, you don't have to move the data. So yeah, so here we find some information about the actual, Google bucket that I have and how much data is inside of that. And then I have links so I can go view that data in the Google Cloud Console. I see the owners of my workspace. These are tags that I have to make it easy to find my workspace. And then different notifications. So maybe I want to get emailed every time I run a workflow and I want to see, the status of it. So I can set up different kind of notifications there. If the data has changed, I might want to know as well. So that's a little bit about kind of the information at the workspace level. Oliver already shared like the CFDE workspace and shared like the ability to, write nice text here, add pictures, really tell people, what are you doing in this workspace? What are you trying to accomplish? And then, when you're looking at featured workspaces, you can also read exactly what people are trying to do in their workspaces. Okay, so you can see here that I have read-only access to this workspace. It's not a workspace that I, own. So if I want to do any kind of analysis using any of the workflows in here or the data tables, then I would make a little copy of it by clicking clone. So I won't do that now, but I just wanted to point that out. If I wanted to do work, how would I do it? That's how I would do it. Okay, so now going over to the data table. All right, so when people are kind of new to working in the cloud, they have a lot of questions about like how do I deal with my data in the cloud? And what are these data tables and how should I think about them? So the data table or the data page is where you can organize your metadata, input files, your output analysis files, sample information, reference information, all of the data you need to do your analysis. So basically the data is, you can organize them in these data tables. And so the sample table, for example, you can see we have our sample ID, that's our unique identifier for all of our samples. And we can see different metadata about the samples and we can see files linked to the samples. So here we can see basically that this file is not located in my workspace's Google bucket. It lives in another Google bucket. And if I actually click on it, I can see more metadata about this particular file. And I have this nice handy copy command if I want to, go into the terminal and just make a copy of the file somewhere. Yeah, so that's that. So these are actually pointers to the files, obviously they're not the files in and of themselves in this data table. So we have different types of files. You can store any kind of file in a Google bucket. So you can see we have, our BAMs, BAM indexes. Some of these are not filled in. And that's because maybe when I was running the analysis, I didn't want to, run it on all of these files. So something that's really cool and an interesting new feature is the ability to see provenance on my data table. So for example, if I'm curious, like how did this column get here? It was configured as an output for the following submissions. So the submission is the workflow that I launched, and here's kind of a unique identifier for that. I can see when it was run, who submitted it, and then that's the file. So that's really nice because when you're, working in your data tables and you're doing lots of different analyses, you might wonder like, well, when did I create this column? Like, what is this, when did I launch that? Like you can then see the history of how your data table has changed. So yeah, so that's a little bit about the sample table. The sample set table is basically a way to group samples. So if I look at kind of the last column here, I can see I have an array of samples that represent this particular sample set ID, and I can see that here. So say I just wanted to quickly look at information about a particular sample. I can search in the table itself to find the sample that I'm looking for, or I can search across the data tables here and look and see, okay, it's available. I can see that link here. It doesn't exist in my sample set. So that's a way, especially if you have maybe like, hundreds of rows here, it might be hard to find what you're looking for. You can search on the individual level like that. So that's kind of the data table section. When you're actually creating a workspace for the first time and you're trying to, create your own data tables, you can actually name the tables whatever you want. This is just an example, a common example of how people sometimes set up their data, I guess, with samples and then grouping them by a certain set of them. But that's a way to set up your data. And then we have the ability to set up and pull in reference data. So if I, want to add like a common genome reference or, other references, I don't have to bring those to the cloud. I can just use what's available here. And then finally, workspace data. Basically, if you have individual files that don't really make sense in a table format and you need to use them for your work, you can just throw them in your workspace bucket, and then you can basically set them up here to be able to use them downstream. Okay, so basically the reason for these data tables, we're trying to organize our information that we want to use in a way that we can view it, and we want to set it up for using in workflows. So before I get to that section, I want to quickly give everybody a chance to think about these few questions that I have for you. And then if you don't know the answer, just stay tuned and I will tell you. So the first question is, what kinds of data do you see in each table or section? Okay, so we talked about it earlier. We have metadata about the particular samples. We have different files and different file types that are linked to the rows. And I can pretty much store like anything in a Google bucket that I want, any kind of file. And I can basically set that up here to, link back to a particular sample. How are these two tables different? Okay, so the sample we know is an individual unique sample, and the sample set is grouping those samples into whatever particular grouping that I want. And this is basically an array of samples in the data table. Okay, and then what cloud platform hosts these files? All right. It's the Google Cloud Platform. Okay, and then if I click on the file, I can see exactly where. All right, so that's our quick rundown of the data tables and the data section. I think I've covered everything I wanted to cover here. So I will move on to the workflows, analysis, and job history section. Okay, so for workflows, Oliver shared, the CFDE workspace and showed that he had different workflows that you can view. So I'm going to just show you another example of that with different workflows. So here I can see information about the workflow and where it's stored. Oliver shared an example of running those workflows with direct file paths, but you can also, and he also showed this example of running your workflow with your inputs pointing to the data table. All right, so this particular workflow is showing it's configured to run with workflows inputs defined by the data table. They're using the sample data table. These are all little, like I would say, more advanced kind of features for running workflows that I won't get into today, but the little eye icon can share with you more about what those each mean. And then here's where I can actually see the WDL script itself, and I can kind of get a better understanding of what's going on here. And then I can see the inputs. So basically what this is saying is for this particular input, for the sample, I'm looking for in the sample table, the SRR-UBAM column, and I want this input value to be anything that's in that column. So let's see, maybe I have to search for SRR-UBAM, but I should be able to find it easily. Just do that. Oh, bad example. But basically that's how that works. So the file or the column name will look for the one that has the name, and it will take whatever is in that particular column and put it as the input. So you can imagine the power there if you're running this on, 1,000 inputs. You don't have to define everyone individually. And then this particular one is saying, look at this reference FASTA file in my Workspace data table. So Workspace is that kind of way of pointing to that Workspace data table. And if I go back, I can go to my Workspace data table and I can see that input there. Alrighty, and then Oliver demonstrated how you can launch your analyses. Oh, I forgot to mention the outputs. So this is where you can define what columns you want to write back to in the data table. So basically it's saying that I want to have all of these different column names, and I want my outputs to be then written to the data table and those with those column headings. So yeah, I think that's all I have to say about the workflows. So we'll move on to what you might be doing after you run your workflows, which is your analysis. So here, this is where you can start what we call cloud environments. Basically, they're virtual machines. You can configure the size of the virtual machine that you want. If you want it to have GPUs, if you want to use Spark, there's different configurations you can set up. So you can power, the machine with how you need it. And then here's just an example where I'm able to read the notebook and see what's inside of it. I'm not going to launch it because this is a read-only workspace. So I'm just going to look at it. But then again, what's really cool about featured workspaces is you can kind of look around and see what other people are doing and, borrow the code from them and not have to, reinvent the wheel there. So that's the analysis tab. And then if I want to start different types of cloud environments, basically this is the Jupyter one, but I would go here and I can set up my different Jupyter, RStudio, or Galaxy environments. All right, and then finally, the job history section was where I can see more information about the workflows that have been run in this workspace, and I can basically view more information about them, and I can go into the dashboard. This is unavailable for me because it's a clone, or sorry, I'm reading it, but then I can see more information about it. There's also a way that you can set it up to show run cost of your workflows, which is handy if you're trying to optimize the cost of your workflow. All righty, I think that is the workspace in a nutshell. The other thing I'll mention is if you click Find a Workflow, wanted to quickly share just a view of DocStore because I think it's pretty great. So we're integrated with DocStore, and here you can see because WDL is a language that we use in Terra, there are 3,716 different WDL workflows that I can look for. And if I'm curious about different organizations that write workflows, I can go look up different groups and see what they have available. If I click on the Broad, I can see different pipelines that are available. If I go to Warp, for example, I can read more about it, and I can actually then view the workflow. And then there's this nice launch with Terra button. So I can take a workflow from DocStore and launch it into Terra. Alrighty. I think that's a little bit of a taste of Terra for you. And I will go ahead and take any questions.
Thanks, Tiffany.
You're welcome.
I guess I can chime in. How, say a user has used some sort of a workflow or something, how do they document changes that have been made to that workflow and things like that? Or how can they see that within the workspace for reproducibility?
Yeah, so documenting changes to the workflow. Let me go to a workspace itself so we can look through it together. So when I think of documenting changes to a workflow, I guess you can document them in a few different places. So my workflow lives in the in DocStore, I can create different versions of the workflow and have different descriptions of what they are. In Terra itself, if I'm going to, I can't click through it this way. Let me go in here. So the Broad Methods repository is actually getting a facelift and will finally be moved from FireCloud over to Terra. So I can click here and then I can see different snapshots. So these are different versions, and I can then, write a snapshot comment and say, this is what I've changed from the previous version of my workflow. And then when I'm, actually running the workflow itself in Terra, I can leave a comment and say that I have a few different versions that I'm testing while I'm going through this. I can leave a comment and say, this is the version, this is what I changed, or whatever comment you want. So that's another place where you can kind of document things that are changing as you're working.
Great, thank you.
You're welcome.
Any other questions for Tiffany? All right.
Thank you, Tiffany. We really appreciate that. But before we wrap up, I just want to share some upcoming events that are going to be ongoing in the Common Fund data ecosystem, especially as, this is normally when we have our public webinar series. So I just want to bring the attention of the webinar series and a couple other events. So first off, as I mentioned, this is our normal time for the public webinar series, which is the last Friday of every month from 1 to 2 p.m. Eastern. So these are the upcoming 2025 webinar dates. So two to look forward to in the upcoming months. February 28th, we'll have 4D Nucleome, and March 28th will be the Knowledge Center. So we will promote the agenda and topics for these webinars a little bit closer to these dates. But for now, if you want to have them on your calendar, these are just some that we are looking forward to as a whole. As part of that, I know we had mentioned this a little bit earlier with the training center. There are a couple conferences that us as the Knowledge Center and various other community members will be attending from CFDE. And so the first is Bio IT World, which is April 2nd to 4th in Boston. I know we had mentioned this previously, but I just want to put this back on there. The second is the ISMB, or the Intelligence System for Molecular Biology, which is July 20th through 24th, and we'll be applying for a series of talks. And we know that there are going to be various groups that will be at this conference as well throughout the CFDE, so we're eager to collaborate on that. And lastly, we will be going to the American Society of Human Genetics, or ASHG, in October, which is the 14th through 18th in Boston. So if you'll be attending these, please take a look out for all of our CFDE events. We'd love to see you there as well, and be happy to participate if there's anything else in the community as well. And lastly, just some other community events that I want to highlight from the various programs and DCCs that are in CFDE. So we have two GlideGen webinar series. This is monthly. Their next one is on February 4th and will feature research collaboration, service, and training at the Complex Carbohydrate Research Center. So that talk will be on the 4th. There's more information on their site. And then in April will be the Bridge to AI Voice AI Symposium and Hackathon. And again, more details are on their site. But I just wanted to bring these to your attention in case you're looking for more events within the ecosystem. So with that, thank you again for taking the time to participate today. We are very excited to hopefully have more of these in the future and feature other programs and DCCs within the CFDE. And again, if there are any questions or concerns, we can be reached at help@cfdeknowledge.org, which I see Julie put in the chat. So we have that there for you. As a reminder, because this is recorded, we will be uploading to our YouTube, which will be sent out to all of the attendees afterwards. I will also pile together all of the slides from the various presenters and share that as well. Please feel free to share with your colleagues if they weren't able to attend. We just want to say a big thank you to all of you, and thank you to all of the fantastic presenters and panelists that we had throughout today. This could not be done without you. So thank you, everyone. And if there's no final questions or thoughts, we'll leave it with that. And thank you, everyone. All right.
Thank you, everyone. Bye-bye.
Bye.